to get going. So just a couple of points. So next week you have your term test. This weekend I'm a little bit busy because they have the Canadian Association for Neuroscience happening here at the Sheridan Center, and I have a number of things that I have to do there. However, I still want to help you get prepared as much as possible, and then during the break we'll discuss how that's going to happen. So the next 2 weeks you actually have it pretty rough, to be quite honest. So you have your term test next Monday, and then we'll do a group activity. It'll be fun. You'll get, everyone who's here will get like the full 1.5%. It'll be great. But the week after, you actually have your review paper due, right? Working on your review papers. I haven't seen very many questions related to your review paper, so I'm a little bit concerned. So hoping that you're staying on top of it. If over the next week or so, so right now concentrate on your term test, but if you have your review ready after the term test, like Wednesday, Thursday, Friday of next week, then you can share it with me as a Google Doc, and then I will comment on it and give you some feedback before you submit it. So get busy, start working on it, start thinking about it. Again, we have a couple of minutes to go. I haven't written the term test yet, so I'm going to be spending time after today's lecture going over and writing the 40 questions that you're going to answer. So you're going to help me a little bit by choosing one of these three numbers. Three. Can we get a consensus? Two? Two. Okay, so I heard an emphatic 2. So we'll do it in reverse order. So 3, you would have had a really easy term test. So not 3. 2. No. Hard. No. You never joke around about. Shall we try it again? Yeah. Okay, so let's try this again. So I need an answer in the next three seconds. Three. Okay, so we'll go with 3. So it's easy, but more like medium easy. So one of the things that you'll find about this upcoming term test, it'll be a lot easier to just read through and get the answers. I'm going to be pointing out specific slides as we go through today's lecture. I didn't finish the last lecture all the way through. You don't have to worry about anything that I did not cover in the last lecture. I've condensed it down to what you need to know on the upcoming slides. And today's slides are the ones that you need to concentrate on. None of the ones that weren't covered last week. Okay, so I'm just going to get started if you don't mind. We have a long 3 hours. There are about 100 slides to get through. Some of them are really detail oriented, and I want to make sure that you understand exactly which slides you need to know, including this slide right here. So this slide, I'd actually expect you to know. You are not going to have a calculation. But in terms of physiology, like all of you who are leaving a physiology program should know that the average blood pressure in the aorta is related to what is known as the mean arterial pressure, or the MAP. And this mean arterial pressure represents the average blood pressure in the aorta. Why is that blood pressure important? Because it's the average blood pressure that is propelling blood throughout your entire body. So what's leaving the aorta, what's getting out of the heart, represents the driving pressure that is moving blood throughout your entire circulatory system. And I need you to know this calculation because it's a really important one. The mean arterial pressure is calculated as the cardiac output. Sometimes you'll see it in your textbook as the total cardiac output And this is the amount of blood pumped by the heart per minute that you calculated, and you were to collect all the blood that's being ejected or leaving the heart after 1 minute, this would be the total cardiac output. If you then multiply this, the total cardiac output, by the peripheral resistance, how much resistance is there, then you get the average blood pressure at the level of the aorta. So think of it really simply. If you have a straw and you're blowing through a straw, and you have a bigger straw versus a straw that has like a really narrow opening, that narrow opening represents having like a higher peripheral resistance. It's harder to blow through a really small opening straw. It's easier to blow through a straw that has a wide diameter. In addition to that, you also need to know that this cardiac output is related to the stroke volume. So this is the blood pumped by the heart during each beat, and we have the number of beats per minute. So if you do all the math, that tells you the number of liters of blood per minute, which is the total cardiac output. I don't need you to know what the cardiac reserve is, but I do need you to know how to estimate what the mean arterial blood pressure is. Okay, we cannot measure this really easily. Some parts of this are really easy to measure, so we can measure your stroke volume relatively easily. We can measure your heart rate really easily. We can't really tell you a lot about your peripheral resistance easily, and as a result, we have to make a guesstimate in terms of the mean arterial pressure. This is a slide on over the weekend to know how this is calculated, what mean arterial blood pressure means, where it is being measured, and how these different elements, stroke volume and heart rate, play a role in all of this. So some of the ways in which this heart rate, stroke volume, and everything else gets regulated are the next few slides. So again, this goes back, how do you actually regulate how much blood the heart pushes out? Some of it is intrinsic. It really depends on how much blood is filling it up. So the heart is a muscle. So the more you fill the heart that there is in it, the harder it's going to push back, and the more it's going to eject out. So this gets into this idea of preload. And this is the intrinsic regulation. This is the heart itself controls how vigorously it pumps out and how much it pumps out. The more you fill the heart, this is what preload is, the more you fill the heart, the greater the stretch is going to be. The greater the stretch is, the greater the contraction is going to be. And that's what Starling's law says. And it is related to preload, how forcefully the heart itself will actually contract. The afterload is the amount of pressure that the contracting ventricles have to produce to overcome the pressure in the aorta. So the heart is not as sensitive to afterload as it is to the preload. Okay, so if you're exercising, for example, and your heart is receiving more blood, bigger, the heart is pushing back on it equally vigor and as much force as possible. So I'm going to be very straightforward with you. I'm going to cover this section. I'm not going to be testing you on these baroreceptors and chemoreceptors. We're going to come back and talk about the cardioregulatory centers a little bit later. But if for example, your cardiac output is controlled by preload and afterload, this goes back to this equation, we can control the amount of blood going into the heart through this cardiac output related to this preload effect. Rate. The heart rate can be affected by things like baroreceptors and chemoreceptors. We can slow down, or speed up the heart rate through these different baroreceptors and chemoreceptors. So again, there's a region within the medulla that is known as the cardioregulatory center, and this will affect like how many beats per minute your heart is beating or pumping. And so there are some reflexes that are involved in this. It's not just how much blood is filling the heart. You can also control the heart rate directly itself, which is what this slide is saying. So if you put all of that together, the cardiac output is regulated by how much blood is returning to the heart. That's Starling's law. And then we can regulate the heart rate. Again, mean arterial pressure is equal to cardiac output times the total peripheral resistance, and then cardiac output is related to heart rate times stroke volume. Stroke volume is this factor here, this preload, and then the heart rate is regulated by this cardioregulatory center. And again, the cardioregulatory center regulation right here on this diagram, I don't need you to really memorize. It's just there to give you an idea that when we talk about mean arterial blood pressure, and I'll just do MBP instead of MAP, that when we talk about the cardiac output times the total peripheral resistance, these are factors that we know get regulated. We can increase these in the stroke volume by Starling's law, and then the heart rate, various regulation on the cardioregulatory center. We can speed up your heart rate. We can increase the stroke volume, and by doing so, we can affect your cardiac output. So what would happen if I increased your stroke volume? What would happen to your mean arterial pressure? It will also increase, right? Would you be able to answer that on Monday? You're sure about that? Okay. What would happen, for example, if I increase total peripheral resistance? Would that increase your mean arterial pressure? Okay. So this is more pathological. It's harder to actually go back and measure this, but we're going to see how that might be measured a little bit later on today. So keep this sort of equation in mind. These are different diseases and disorders that you might be interested in if you're following up on this section, if you're really curious about this section, where there are different aspects. Some of these are genetic, so we have problems where we have reduced blood flow. That's something that is not genetic. That's something that you guys are all doing to yourselves. By not exercising, not eating healthy, you are going to be damaging your heart muscle. There are congenital heart diseases that are more genetic in origin, where you might have a septal defect. There's a hole in the wall between the left and right sides of the heart. And if you're really interested, this is sort of a great place to go back, see what types of disorders of the heart that you might be interested in following up on, and you can go and study that more independently, come back and chat with me about any of these different disorders that might be of interest to you. So I'm now going to switch gears. Slides that I need you to remember from the end of the last lecture. So I didn't finish all of the last lecture. There were some slides that were left over. I went through the most important ones. The ones I didn't cover at the end of last lecture, you don't need to worry about them, okay? Only the slides that I covered. So if you take a look at the circulatory system, we're not talking about the heart anymore. We're talking about the rest of the vessels associated with the heart. The functions of this circulatory system are obvious. Things to carry blood. We'll see how this is important in the respiratory system later on today. By carrying things in the blood, we exchange nutrients, waste products, and gases, especially O2 and CO2 that you'll see in the respiratory system. We also use this to transport hormones that we've talked about. We're not going to have much time to talk about the immune system, but the blood is also there for things like coagulation, etc. And it's also there to regulate blood pressure. And we're going to spend some time on blood pressure in the next few slides. I don't need you to memorize this slide. I do need you to think about this slide. I don't need you to memorize the numbers. I'm not going to ask you on a multiple choice test question, like what percent of the blood at any one given point in time is contained in the heart. 8%? 10%? I don't like those types of questions. But I want you to think a little bit about this. This is where you are looking at your blood at any one given point in time. Right now, when you're sitting there, most of your blood is where? So most of your blood is in the veins and venules. Surprising? Not surprising? Why is it not surprising? Blood flows slowly? Through the veins? But they're all connected, right? Are they not connected? Don't they all make a circuit back to the heart? So why are they all there? Because the title actually tells you these are capacitance reservoirs. Do you know where we get the word artery? So imagine you're back in the 17th century. It's the mid 1600s. You're William Harvey. You're doing a dissection on the human body. You cut open someone's arm. You expect blood to be gushing out, but in fact you don't. You see these vessels, and when you cut these vessels, in a living person, when you cut these vessels, blood starts squirting out. It's red, it's oxygenated, it's really bright red. But William Harvey, when he's examining the circulation in the 17th century, in the mid 1600s, he sees these blood vessels. When he cuts them on a cadaver, what does he see? Arteries. Why call them arteries? Because they're full of air, and that's what he called them. In an individual, the vast majority of your blood is actually pooled in a living person in the veins. In a cadaver, we don't see very much blood left at all in the arteries. They're pretty much empty in a cadaver, and this is why they called them arteries. And even in an individual, a living individual, right now, if you're not exercising, the amount of blood flowing through your systemic arteries and arterioles is only 15%. Even though you see in the movies that if you cut an artery, you see like blood squirting out, overall, there's very little blood actually contained in the arteries or arterioles. So think about that. And I want you to not memorize these numbers, but to memorize the overall distribution. We don't have a lot of blood in the capillaries, nor do we have a lot in the arteries and arterioles, but we have an awful lot in the systemic veins. Why is that important? Because the minute you start exercising, that blood gets pushed from here into your arteries, back into the heart and everywhere else. The distribution of this changes. So I'm going to take you through. So that previous slide was important as a concept slide. I want you to know the overall proportions. If you're leaving a physiology course, you should know that the majority of your blood, when you're not exercising, is actually sitting in your veins, not within the arteries. But we are going to talk about some of the features. Why is that the case? Why do we have so much blood in the veins and not in the arteries, right? This was the question that I asked your colleague. So there's some very basic differences. The arteries are elastic. They have a lot more elastic tissue associated with them. They are much more muscular in terms of the walls of the vessels that we take a look at, and they're also connected to these arterioles. The capillaries are going to be the site of exchange at the one cell thick level, and the veins that we just talked about, the ones where we actually see the vast majority of your blood being pooled, 60% of your blood is in the veins and venules. One of the reasons why is they actually have thinner walls. They have less elastic tissue, and there's less smooth muscle. The arteries are great reservoirs for generating pressure because they're elastic. Like as soon as blood hits it, it squirts it forward. But the veins are actually really good at pooling and keeping and storing blood there because they're thinner walls, they're not as muscular, they're not going to push blood through, and they're overall less elastic. And this basic property is something that I want you to be able to tell me a little bit about. Any questions about that before I go on? No? Okay, so this gets into the smallest level. So if we take everything from the aorta through the systemic arteries to the arterioles, we're eventually going to get to these capillaries. Capillaries come in different flavors. And again, as physiology students, you should make sure that you know these different types of capillaries that I'm going to go through in the next slide. I want you from this slide to be able to tell me a couple of things. One, that the capillary walls actually consist of these endothelial cells. Endothelial cells are the same lining, by the way, that make up the heart wall. So endothelial cells in the heart wall, endothelial cells in the arteries, arterioles, capillaries are no different. There is a basement membrane that surrounds them and then connective tissue. And we might have some other cell types like fibroblasts and macrophages, but overall it's really just an endothelial cell lining. Those are the cells that we find within these capillaries. And different substances can move through these different capillaries. If they're lipid soluble or water soluble, they can just go directly through the plasma membrane. And larger water soluble molecules will go through openings that we're going to call fenestrae or fenestrations, which mean like windows. And if you studied grade 9 French and you learned about a window, la fenêtre or something, le fenêtre, whatever it is. I didn't do very well in French either, you can tell. These fenestrations or windows are gaps that exist between endothelial cells that allow larger molecules to go through. That's the general way in which all capillaries look. However, And this is a really busy slide. And then there are specific diagrams of this on the following slides. But this is a slide that I want you to spend some time on in the upcoming few days. You are going to get a question, and I guarantee you're going to get a question related to this slide and the different types of capillaries. And part of it is really because capillaries are important, and also knowing like the basic principle about why these different types of capillaries exist. So you might, for example, have these continuous types of capillaries. There are none of these fenestrations. So again, you should know the basic features. Continuous capillaries have none of these fenestrations. Therefore, without these fenestrations, they're less permeable to those large molecules. Where would you see these types of capillaries? We've already talked about the blood-brain barrier. Remember, they're really tight, the tight junctions. You don't get things into the brain easily. And so in nervous tissue, in places like the brain, we would see lots of these continuous capillaries. So where you'd see it, make sure that you know what continuous capillaries are, and also within the muscle. Fenestration means that they have gaps or pores, and these endothelial cells have a lot of these different types of fenestrations or pores. So again, where might we be able to see these really permeable types of capillaries? Well, one of the places that we'll see eventually is in the kidney. We need to be able to filter your blood at the level of the kidney a lot, and we're going to see that really soon. The only way that you can filter things in your blood is to have capillaries that are dumping things off into the kidneys that allow different molecules to pass into the kidneys. The only way you can do that is to have these fenestrations. Okay, so there are other examples as well, but the main one that I want you to make sure that you know are the glomeruli of the kidney, and also the small intestinal villi. The other ones, like the choroid plexus and the ciliary process, you don't need to worry about. Villi and glomeruli. The sinusoidal ones, again, these are larger diameter capillaries. These also have large fenestrations. Overall, they tend to have less basement membrane, and we might see this in an endocrine gland. Okay, so you might see this, for example, in the adrenal gland. You would see this type of capillary supplying the adrenal gland. And again, I'm not going to try to confuse you too much. There's another subtype of this known as the sinusoids. I'm just going to put this up here for completeness if you want to go back and examine this yourself. I know some of you are really somewhat anxious about your upcoming term tests, so we'll keep it really simple. You are going to get this on your term test. It is a really important concept about how these different capillaries work, why they work in the way that they do. You should have some physiological sort of rationale for why they're in these different tissues, but I'm not going to try to make it too complicated for you. After all, you did pick the number 3, right? So the sinusoids I'm just going to avoid for the time being. We will see this later on in terms of the liver and a little bit in the bone marrow. And then the venous sinuses, again, I won't worry about those either. So that's really the continuous, fenestrated, and sinusoidal that you should worry about. They're all somewhat related. They all have these gaps and these pores. They're just different in terms of size and number in different types of tissue. Any questions of what you need to know here before we go on? It makes sense in terms of the physiology. Later, when we talk about the kidney and you remember these fenestrated capillaries, it'll make sense to you, like why things get filtered through the kidney, why we need these holes in the blood vessels going into the kidney. Okay, so again, this is just sort of a diagrammatic view of the different types of capillary walls. This is that continuous capillary that we mentioned. We would see a fair bit of within the brain as well as muscle tissue. You don't have to worry about fenestrations with or without a diaphragm associated with it, and the sinusoidal capillaries as well, not to spend too much time on it. It's just there for your interest in terms of what these might look like in cross-section. Okay, so this is actually a really easy to understand slide. I don't need you to memorize the microanatomy that you see here on the left, but this becomes really, really important. for a couple of reasons. I don't know if we're going to have time to come back and talk about allergies and histamine and other types of things. But one of the things that you see on this particular diagram is that the arterial system is actually continuous with the venous system. So the arterioles and the venous system are actually interconnected by these capillaries. So it's not like you have the arteries, they stop, and then you have another set of veins and venules that begin. They're all interconnected. One system is oxygenated and the other one is deoxygenated in blue. These are really important is if you look very carefully at the smaller arterioles, which we call the meta-arterioles at the very top of the diagram, these meta-arterioles have on them these smooth muscle sphincters that can open and close. So this regulates like how much blood is actually flowing through a particular organ. You can turn off a whole branch of the meta-arteriole. Imagine, for example, instead of blood flowing all the way through like this and then reaching the venule, imagine for a second that you closed off all of these arteriolar sphincters, these meta-arteriolar sphincters. No blood flows through here at all. Okay, so that means that most of the blood is flowing through here and then it might go through this direction. But imagine for a second, and this happens all the time, like you're sitting here right now, some of your meta-arterioles have collapsed, the sphincters are closed, you're not feeling the effects of this. But imagine for a second that you opened everything up, like everything is opened up at the same time. So some are closed, some are open all the time, about 50-50 actually. But now everything opens up. That becomes a real problem. Like you won't have the same kind of cardiac return because the blood that normally should be going through at a certain rate, because some of these are open and some of these are closed because of these smooth muscle sphincters and precapillary sphincters, now is just flooding all of the little capillaries along the way. And that becomes a problem. We'll talk about it when we get into the immune system and what happens when you have allergic responses. And we'll come back and talk about this a little bit later. For the time being, I need you to remember that these smooth muscle sphincters control blood flow at the level of the meta-arterioles and in terms of these precapillary sphincters. They open them and they close them. Okay, so then getting into these the particulars of the structures associated with these arteries and veins. Arteries and veins, all of them, whether it's an artery or a vein, have these different layers. So there is the tunica media, and then there is the tunica externa. It is also called the tunica adventitia. So arteries and veins have all three layers associated with it. Some of these layers and the functions of these layers, and I am going to ask you to memorize this slide, to go back and review this particular slide. This tunica intima is the innermost layer, and this has an elastic membrane associated with it. The tunica media for us is really important because this is where, when we talk about vasoconstriction and vasodilation, it is this layer, the muscle layer, the smooth muscle layer that's found in arteries, the smooth muscle layer that's found in veins, that actually contracts or relaxes. Okay, so when we talk about vasoconstriction, it's the tunica media that's being affected. It's not the intima, it's not the externa. And then the tunica externa, or the tunica adventitia, is related to connective tissue. And again, it's the surrounding connective tissue on the outside most layer. And again, you don't have to worry about a diagram like this showing up. These are the innermost layers down here at the bottom. This is the outer connective tissue layer shown up here in sort of this whitish color. That's connective tissue. The media, again, the most important thing that you should take away, this is where the smooth muscle is found. This is where vasoconstriction occurs and vasodilation. I want you to know that basic principle. Do we find this in arteries or veins? We find them in both, correct. So both in arteries as well as in veins. It's the overall structure. So why do we talk about arteries and veins? What was different about them? Sorry? Nope, not direction of blood flow. Yes.
Arteries are better at pushing blood through, whereas veins are better at pulling blood
Sure. So this gets back into when we look at arteries versus veins, arteries are high pressure reservoirs and then veins are capacitants. They store blood better. And this goes back into there are differences in the layers of tissue that we find here. And this is really evident here. If you take a look, this is a cross section through the skin of an individual. This is at 250 times magnification. These are red blood cells in the middle. This is the lumen. This is where red blood cells are normally flowing. This is the cross section taken through an artery, A, and in B, and over here, this is the cross section of a vein. So a couple of things, not the same. They have all three layers. They have the intima, the media, and then the adventitia or the externa. But you can see that the overall proportion, the size of these different layers is actually quite different, especially this layer that you see right here. The tunica media, this layer is much, much bigger than it is over here. Muscular, smooth muscle, it's involved in vasoconstriction and vasodilation in the artery rather than in the vein. And this gets into the elastic artery. It has more elasticity associated with it, and it also has more elastic tissue overall than muscle. And again, this type of artery is relatively thick, and this is just one type. There are many other types. That previous diagram is a different type. This elastic or conducting artery has a relatively thick tunica intima and a relatively thin tunica media that you see here in the middle. Okay, so again, as a general principle, there is a difference in the different types of layers. So that was the elastic type. Again, don't worry about the details here. I'm going to point out what you need to know because there are many, many different types of arteries. In contrast, this is a muscular artery, or also called a medium artery. This has much more smooth muscle. It's there to regulate blood supply. This is the one that vasoconstricts, vasodilates. These are the ones that are adapted specifically for that. And you can see that their overall proportion of the tunica media is different. Okay, and this is most of the smaller arteries that you have throughout. And this is the one that I want you to remember can regulate vasodilation and vasoconstriction. And that's all I really want you to know. I don't need you to tell me the difference between elastic and muscular. The main thing is when we're talking about arteries, they're the ones that get vasodilated and vasoconstricted, and they do so because they have the smooth muscle that's capable of being vasoconstricted and vasodilated. Any questions before we go on? Okay, so big principles, some detail related to the capillaries. The big principles being there are differences between arteries and veins. You are responsible for knowing the different layers of the arteries and veins, starting from the inside, which is the intima. The media is the next layer, and then on the outside, we have the externa. And what was the other name for the tunica externa? We really have to work on your short-term memory. Yeah. Yeah, the adventitia on the outside. And so if we're taking a look, those are the ones that I need you to know for specific details. Capillaries, you need to know like the fenestrated, the continuous type, where you find them, what their major properties are, and then the overall idea that veins are capacitance reservoirs versus arteries being pressure reservoirs. Any questions? Can I go on? Okay, so these venules, again, help to drain the capillary network. They consist of endothelial cells. This whole vascular tree, it's endothelial cells on the inside anyways. There's always a basement membrane, and overall, even though it says the amount of smooth muscle increases, I hope I've convinced you that overall, there isn't a lot of smooth muscle compared to the arteries. Okay, so smooth muscles, again, form this continuous layer. So if you are taking a look at the veins, and one of the reasons why we have these pressure reservoirs, this capacitance reservoir, and your colleague was mentioning that the blood doesn't flow as quickly through them. In a way that's actually not inaccurate. So unlike the muscular arteries, so you can imagine like if blood is rushing through and you have like an elastic push behind it, it'll be ejected forward as long as that elasticity pushes that blood forward. I told you there's not as much smooth muscle and elastic tissue within the veins. And so how do you move things through here? It's much more challenging. And within the veins, especially ones that are larger than 2 millimeters across in diameter, we have these specialized types of valves. And what these valves do is that every time the muscles in the tissue that these veins are embedded within, as the muscle contracts, so as you're walking, the veins in your legs are being pushed open, the blood moves forward, and then when your muscle stops contracting, these valves close behind it, so the blood gets trapped here, right? The next time that your leg contracts, the blood moves up. It seems really inefficient, but because the veins are actually designed to hold blood, it's actually not inefficient. So these valves are the ones that I'd like you to make sure that you are aware of, are found in veins. We have more of these veins in lower extremities, in other words, in your legs and the bottom parts of your bodies than you do up in your arms, which are your upper extremities. Can you think of a reason, consequence, where your valves fail and it leads to a consequence? Yep. Bulging veins, like this bulging vein. But maybe those aren't veins. Could be arteries. Like if they are you talking about someone who like works out, goes exercising, jogging? No? Anyone else? Yes. Yep. Yeah, so what you're talking about are varicose veins. Have you guys heard of varicose veins? And what happens is some of these valves just don't work as well as they should be. The blood gets trapped here, and it kind of looks like a blue color if you're looking at someone who has varicose veins. The only way that you can actually get rid of them is to pop a needle into the vein, drain it of blood, and then the varicose vein disappears. And that's, again, physiologically one of the reasons why these valves are important. We don't want them to fail. We want them to keep pushing blood upwards. But in older individuals or individuals who have orthostatic hypertension because they've been standing all day, it doesn't work. These things all have some consequences associated with it. Again, I'm not going to be asking you about the innervation, the neural innervation to these blood vessels, other than knowing that this vasoconstriction that we're talking about really occurs at the level of these arteries and arterioles. And I mentioned this already, and this is that vasoconstriction, and they have the greatest amount of this vasoconstriction through these sympathetic nerve fibers. And again, you don't have to worry about myelinated fibers, you don't have to worry about baroreceptors. It's really this vasoconstriction and this sympathetic system that's more important. Any questions before we go on, or any comments before we go on? So normally I don't ask you to make sure that you know definitions, but I do want you to know this definition because I hear students use these terms interchangeably, and for the most part, everyone understands these can be somewhat related. But when you're talking about the aging of the arteries, and you guys are, if you're in your 20s, then you have nice healthy arteries. But as you get older, no matter how healthy a lifestyle you have some issues that happen. And one is arteriosclerosis, and this is a general term for degeneration in the arteries, making them less elastic. So in other words, everyone has arteriosclerosis. Your arteries just aren't working as well as they used to as you get older. Not everyone, though, atherosclerosis. Atherosclerosis is related. The blood vessels don't work as well. The arteries don't work as well, but they're not working as well and not transmitting as much blood through because you start depositing fat or plaques on the vessel walls, right? So normally It starts off sometime in, as far as we know, sometime in your 20s, early 30s. You get this little nick in your blood vessel, in your artery. You get what's known as a fatty streak. It actually looks like a little yellow streak. That's all it is at the beginning. And then over time, if you don't have a good diet, you will start accumulating on that little yellow streak more and more of the fat deposition occurring, and then you'll start building up this atherosclerotic plaque on your blood vessel wall. So I want you to make sure that you know the difference between arterio versus atherosclerosis. Okay. So we're going to go through this section. There are some really key points that I need you to make sure that you know related to arterial blood pressure. This is where you are measuring the systolic pressure. The systolic pressure is representative of the ventricular contraction, and the diastolic pressure, the lowest arterial pressure that is exhibited by the heart. And I also need you to know this definition, which is the pulse pressure, the difference between systolic and diastolic. You guys have all had your blood pressure taken, right? What's your blood pressure? Okay, so if you're a perfect physiological specimen, you have like the perfect 120/80, right? So what's the systolic pressure here? So the systolic is the representation of the ventricular contractile force. That's the maximal amount of force leaving the heart at the level of the aorta. And then the 80 is the diastolic, and this represents the arterial, the lowest arterial pressure that is possible. Can you tell me, without looking back at your notes, what is the pulse pressure here? How many of you think 30? 40? 40? 50? So it's just the pulse pressure is this minus this. So that represents a pulse pressure of 40. So just hold on to that thought. That's the only sort of math that you need to know right now. And I'm going to ask you to make sure that you know the definitions of what a pulse pressure is. It actually becomes important in a few. This also gets into this idea. And again, I don't need you to go too crazy on this. All I need you to make sure that you know on this and the next diagram is that normally your blood is flowing through your blood vessels, including your arteries, veins, whatever, in a laminar flow pattern. So that means that it is streamlined. The blood vessel, the interior of the blood vessel is smooth, and the blood is flowing through. It's not making any noise as it's going through. Turbulent. So you've heard of like air turbulence, where your plane is jumping up and down. So think about it as, what would cause turbulence within a blood vessel? Well, this is where, for example, the rate of flow is being disrupted. And we don't normally have that. Okay, if you are a normal, healthy individual, all of your blood flow has to be streamlined. And I asked you this question a while back. If I took a stethoscope and I put it on one of the arteries that you have on your arm, so I identified an artery on your arm, I put a stethoscope on there, if I heard something, that's an issue. Everyone has laminar flow. I can't hear anything through a stethoscope. If you have turbulent flow through that artery, for whatever pathological reasons, there's an occlusion there or something else, then I can hear that on a stethoscope. Normally does not happen. It's not really physiological. And this turbulent flow is partially responsible for those heart sounds, which reminds me, because I told you I'd try to give you as many hints as possible to focus your studying this weekend. Do you think you have to know the heart sounds lecture? Yeah, you do have one multiple choice test question. You should know what causes the lub-dub sound, what's opening, what's closing, how it would be measured like on the surface of the chest. And again, this is related to this turbulent flow. So this is what it looks like. This is normal blood flow is laminar. It's going along in sheets. You can't hear anything. There's no way that it's causing any blood is bumping up against the side of the blood vessel wall. But if you have turbulent flow, where, for example, instead of it being the same diameter all the way through, now you might have like a little kink in this, and then you will see that as it's hitting this sort of neck, that you'll see this blood kind of hitting the sides of the walls and causing turbulence. You'll be able to hear that turbulence. So normally, do we have laminar or do we have turbulent flow? Healthy individual. So it's always laminar. And again, I know how much you really love calculations on your tests. So we talked about cardiac output, stroke volume times heart rate, and again, mean arterial blood pressure, cardiac output, or heart rate times stroke volume times what? Total peripheral resistance. So here's another calculation for you. And again, you don't have to worry about having a calculator. You don't have to actually solve for X or whatever it is. If we're looking at this, you have just measured the arterial, you've just measured the systolic and the diastolic pressure. So you go in, you get a blood pressure cuff. They will measure the systolic pressure by listening for sounds. You don't hear any. When you hear the sound, when the sounds disappear completely and you've occluded the blood vessel, no blood is flowing through, you get the systolic. And then when you start loosening up the pressure cuff and it goes less and less tight and it gets lower and lower, then the first sounds that you hear, just as the blood starts to flow, you hear tapping like on the blood vessel that you have the stethoscope on. That represents the diastolic pressure. So if you take a look at the difference, the systolic pressure and the diastolic pressure. The systolic pressure is the ventricular pressure, and then this diastolic pressure is the lowest possible arterial pressure at the level of the aorta, which is what you're seeing here. One of the things that we can calculate is what is known as this mean arterial pressure, right? So I told you that we can calculate it in different ways, like the mean arterial pressure or mean blood pressure. We can also calculate it this way. So this calculation is a little different. And I need you to make sure that you know that here we are talking about the diastolic pressure plus 1/3 the pulse pressure. And you're going to be asking me, but what the heck is a pulse pressure? Do you remember what the pulse pressure is? So it's the difference between the systolic and diastolic. So if I were to give you a value like 120 over 90, and I asked you to calculate the mean arterial pressure, would you be able to tell me what that is? Yes. Okay, good. What is it? So let's just start with what is the pulse pressure? So 120 minus 90 is 30. And then we take the diastolic. And which of these is the diastolic? So we take 90. And then we take 1/3 of the pulse pressure, which is, again, 30. So what is the mean arterial pressure? 100. So 90 plus 1/3 of 30, which is 10, is 100. So even if you don't have to calculate it all the way through, I'm hoping that you'll remember at least this calculation, the diastolic pressure plus 1/3 of the pulse pressure. And overall, as you get farther and farther away from the heart, all of these different pressures eventually disappear. We don't have a lot of pressure in the ventricles. We already know that. Don't have a lot of pressure in the veins. We already know that. Shows you in a different way that the arteries, arterioles, are where you have a pressure reservoir. And here we have very little pressure, but this is where blood gets stored, okay? Any questions on this before we go on, or any comments, any other sort of things that you want to discuss before we continue on? I have a few more slides and then we'll take a break. Again, I don't need you to memorize this either. We're going to have to talk about this when we get into the kidneys. We get very little pressure. So we start off with like 120 millimeters of mercury at the level of the ventricle as it leads into the aorta. But by the time we get to the capillaries, we have like almost nothing. We have like about 15 to maybe 35 millimeters of mercury in terms of pressure. That's actually good because remember, what is the cell lining on these capillaries? So endothelial cells. How many endothelial cells? So it's one cell thick. if we had really high blood pressure, those capillaries would burst, and that happens. Like if you ever see someone who has like really high blood pressure because their diet, they have a lot of sodium, or they've engaged in like really strenuous activity, you actually see the blood vessel bursting, like in their eye, for example, and that also represents bursting of blood vessels, capillaries within their body. You just can't see it. Like they are bursting in there too. You just can't see it, but you can see the ones in the eye. And this is because high blood pressure actually causes these to burst open. And again, we won't worry about these values or anything else. We'll come back to it. This is not a slide that you need to memorize. This is a slide you need to review. Again, the venous blood pressure, you don't need to worry about this either. There isn't a lot of pressure on the venous side of things. We know that the arterial side is where we have the greatest amount of pressure associated with it. I've already talked to you about these things related to contraction of skeletal muscle, these valves that we talked about within these larger than 2 millimeter veins earlier. And this is that idea of a muscular pump that pushes blood back toward the heart. And if these valves, for whatever reasons, fail, they stay open, blood pools here. You actually see the pooling of blood in the form of varicose veins. Okay. And have any of you actually seen varicose veins being removed? A needle, they pull the blood out of the veins that have these faulty valves, and then the next time that individual has that again, they have to go in and get that blood pulled out as well. So I don't need you to memorize all of the things that are on this particular slide. I told you also not to worry about vasoconstriction too much here either. Any questions before we go on? Okay, so I'm going to switch gears now. That's the end of the cardiac system. And again, if you're thinking about this, you'll get 40 questions. It's roughly going to be about 22-ish questions based on the cardiac system that we talked about. Sorry, the muscle and cardiac that we just finished. About 22 to 25 questions, I'm assuming. I'm told that I should be giving you easier questions, right? No? No, or was it harder? I can't remember. I think it was harder question. It was easier. Okay. And then the remainder will come from this part of the lecture. So again, I'm not going to have time to get into the theoretical perspectives on why we breathe, but we are going to go through some of the different functions of the respiratory system, some of the anatomy of the respiratory system, and to point out what you need to know on the slides as we go through. I want you to, as we go through those slides, circle those slides because there'll be questions taken directly from those slides, and that's the way I like to form my questions. So if you didn't eat, you would actually not have to breathe. The only reason you breathe is because you eat. Go talk to your biochemistry prof. They'll verify this for you. The only reason why we actually do this is because of the electron transport chain. The only reason we need the electron transport chain is because we need to generate ATP. The only way that we can generate ATP is because we need oxygen sort of sink electrons as they move along. So we'll eventually come back, talk about eating and talk about digestion a little bit later on. Something to keep in mind. There are lots of different functions for the respiratory system. Don't try to memorize the slide. There's a lot in this upcoming section, and there's some concepts coming up in this section that we need to get through. But this involves part of the circulatory system that we just talked about. Of course, you know about the O2 and gas exchange with CO2 between the blood and the circulating air. And we're going to spend some time on this third last point here at the bottom. We're going to talk about some of the respiratory surfaces and the anatomy of the respiratory surfaces. Unfortunately, we're not going to have time to talk about how you actually need to pull air in order for you to generate sound. Your voice, for example, wouldn't work unless you had a respiratory system. But unfortunately, we're not going to have time to do that. And this is sort of a diagrammatic view of the same thing. And we're going to be talking about all of these systems. And when we go through, I want you to these basic principles of pulmonary ventilation, how the changes in pressure work. Then we're going to talk about mechanisms of O2 and CO2 exchange, and then how we transport oxygen and CO2 in the blood. And you guys have all learned about hemoglobin, right? So what can you tell me, just out of curiosity, before we get to that section? Tell me everything you know about hemoglobin, then we'll take a break. Okay, oxygen binds to hemoglobin. Great. What else can you tell me about hemoglobin? Sorry? Changes from R to S states. Okay, what else can you tell me about hemoglobin? Yes. Okay, it does. Sure, absolutely does. Does hemoglobin have affinity for carbon dioxide? So by the end of today, you should be able to give me a more definitive answer for the overall binding of oxygen versus CO2, why we actually need hemoglobin. So your blood, the liquid part of your blood actually is not very efficient in transporting oxygen or CO2. It doesn't dissolve very well in fluid. So we need a way to do it, and this is our solution, is to have a molecule known as hemoglobin to do it. 4 subunits, as you mentioned, related to essential iron molecule. We're going to go through all of that later on today, and we'll talk about the circulatory system. So I think now is actually a good time to take a break. So why don't we take a 10-minute break now, and then I'll take you through different components of the respiratory system.
I got a little confused when you were saying about the pulse pressure. So is that 120 over minus 80, or how would I?
It's always systolic minus.
Okay.
So in that particular example, would be 120 minus.
Minus 80. Okay, and then right. So I have to switch. Okay. So all right.
So if you
Can I go back and try that? But I mean, they're fairly straightforward.
I chose one of the gene editing ones with the muscular dystrophy. And I just have a few questions about, I think I know what it's talking about, but I'm not entirely sure. This one that I chose is talking about 3 different types of like insertion, of delivery. You know what I mean? So I just want to like double check with you and make sure that I know what I'm talking about. Want to go do what? No, I know that they did do different modes, but and I know they compared the efficacy of all 3. But I wasn't sure. Yeah. Yes, that's what they did. They did retro and yeah. Yes, that was yeah, the lining or something. I wasn't quite sure when you were in the chair. Hold on a second. Hold on a moment. Just give me a second.
So what do you understand about that?
Well, I know that this in the this one. That's good, right?
There's a really large known
Yes,
district.
and that's what the reason
It's difficult.
it's difficult.
Or acres, the circular complex that's used in muscle contraction. So in these individuals, the dystrophin complex is gone
At the cell surface.
at the cell surface.
It connects with the cytoskeleton.
Yeah, Yes. So this is that molecule. There's 15 instruments again.
Okay.
So one of the things that's really important is that I think it's exon 52.
This is exon 20.
So what happens is you have all of these different exons. So let's say this is exon 1, 2, 3, 4, all the way there. And then what happens is in this particular exon 23, you might have a mutation here where normally it's not treated. So you have an insertion it's not there. So what happens is you get this truncated version of this work really. However, because of the structure that region that normally is here is actually repeated several times. So it increases so exon 23, the coding region of exon 23 is very similar to one that's coded by exon 24 and very close to exon 22. So it's like having like an extra pair of glasses. You don't really need the extra pair, you just have it sort of as a backup. Normally you have it, but what might happen is if you simply didn't have 23, 22, and 24. It goes on
kind of normally.
Yes. And so what happens here is that the stop codon produces this truncated protein.
Right.
And you manage to get exon 24 to be joined together.
Right.
With this stop codon,
I wasn't sure.
I'm sure that it's a paper that talks about exon skipping.
I don't remember that.
So this gene, this exon actually gets skipped. So
Yes,
it's
cut out.
So that's exon skipping. And what happens here is that if you exon skipping, you get a chain recovery of dystrophin. And what happens is, how can you get rid of exon 23? Well, one of the ways that you can do this is using enzyme to actually cause what is known you cause indel. Talk about
Yeah,
this is an insertion that's put in CRISPR-Cas9 or a deletion.
it's a deletion.
Okay, so whatever actually it causes both CRISPR-Cas9 causes insertion and deletion. Always check for indel formation. They have to check that. When you do this though, what ends up happening is, so what happens when you cause like a indel in exon 23? So what happens is indel gets recognized in molecular biology of drugs.
Oh, I looked that up.
Do you know what monomer is?
No, I had to look it up myself.
So you have damaged DNA.
Yeah, yes. Right, yes.
This is what's known as homology-directed repair, HDR.
I think it was better than that, right?
Yeah, it's different, but the same idea. So you have basic sequence and
I don't know, that's
generally, yes.
But, so it's not like I criticize the paper.
Yeah, you should. That paper is a really good paper, but you should also bring in the two other papers that we
Yeah, the other ones, yeah, I guess they're like
what
difference between the three?
Yeah, they are.
Or,
And then what is the advantage of is the MDX?
I don't remember. Oh, yeah, the MDX one,
Is
yeah.
the MDX mouse a really good mouse model?
Okay. Okay.
They use the right page. Really quick question, really quick.
Okay, so I emailed you 2 days ago regarding this article. It's not really related to physiology, but like I said in the email, it talks about prostate cancer, it's related to that. But then, yeah, your
Don't worry about the rubric. That paper is fine for company.
I was going to email you about the cat paper that I couldn't download. So I was able to download it, but it doesn't have the entire. I don't know why.
Let me recall you mentioned this. I'm gonna try to find it in the website.
Okay.
Okay, if you guys don't mind, I'm going to get going. We have a lot of slides to get through in this section as well. Okay, so in terms of the components of the respiratory system, you don't have to memorize all the details on this particular slide. There are some major points that I do want to point out to you. That there is a change between the upper. Sorry, I can't shout over top of you. So if you guys don't mind, get going. Luca, is it okay to get going? Thanks. Um, So in terms of the upper versus the lower respiratory system, there are some key changes. We won't really worry about the upper respiratory system. The one thing I wanted to actually point out, a lot of you will go in and get a medical note from your doctor that says that you have a lower respiratory infection. That doesn't mean, for example, that you actually have infection down here at the level of your lungs. That's actually really catastrophic. That means that you would have something like pneumonia. What it means, though, is that below the level of the larynx, you might have like inflammation, which means that your trachea might be inflamed. It might mean things within one of your bronchi, but it doesn't mean that your lungs themselves are infected. And we'll talk about this as well. And it's really the pneumonia in the lungs that becomes important clinically. And then the soft tissues that we'll talk about. There are soft tissues throughout the lower respiratory system. Within the trachea, these blue cartilage rings that you see here help in structural support. Can you think of what cartilage is doing in this particular case? Why have cartilage? Do you need to breathe all the time? It's not a trick question. Yes, you do. So how do you get air from your mouth and nose down into your lungs? It's through this opening known as the trachea. It would be disastrous for you if your trachea actually collapsed. It is designed not to collapse by having this structural support around it. That doesn't mean that lower down here in this part of the respiratory tree that it's not impossible to have collapse. In fact, we're going to actually see where this occurs. So again, we won't go through the upper respiratory tract up here. We won't talk about the different structures. I'll just mention in passing the larynx, because the larynx is there to help provide an open airway. It's a patent airway. So if you are really interested in medicine and you're really interested in anesthesiology, then one of the first things that you learn is about airway management. It's the first part of the ABCs in anesthesiology. It's always taking care of the airway and making sure that it's open. And part of this is to make sure that the larynx isn't being blocked. You have an open or patent airway, and this makes sure that you get air into the lungs and food is going into the digestive tract. It's also there for the production of your voice. And the reason why we get food into the digestive system and air into the lungs is because of this epiglottis, which is an elastic cartilage that covers the laryngeal inlet during swallowing so that you don't actually accidentally get food into your lungs, which again would be pretty disastrous. Like getting fluid in there, not bad, but getting a piece of food trapped in your lungs is actually not trivial. So again, we won't talk about the vocal ligaments at all. These are other elastic fibers that you see here on this particular diagram. And I'm not going to be testing you at all on speech and how we open and close and vibrate the elastic tissue on these vocal folds that are part of this larynx. Again, it's there for your own information if you are really curious or really interested in this. And again, you can sort of see that little glottis and the epiglottis up here at the top that will cover this opening during swallowing. Do you guys know the Valsalva maneuver? I think we talked about it. Valsalva? Vocal folds that I just showed you here can actually act as a sphincter, prevent air passage from going through. Valsalva maneuver? Anyone? So this is the maneuver that you make when you go number 2. You kind of close the glottis, you push down. In order for you to do this, the glottis prevents exhalation while you're doing number 2. Your abdominal muscles contract, intra-abdominal pressure rises, and then it causes anything within your digestive tract to be expelled as a result of this increase in intra-abdominal pressure. But this Valsalva maneuver, actually, if you have a really bad heart, can cause issues because it increases the blood pressure temporarily. And so we try to avoid any blood pressure increases in individuals that have any kind of issue surrounding their heart. So even going number 2 with a patient that has heart problems is taken very seriously in the hospital, and they manage everything that they eat. In case you're wondering why people stay in the hospital, it's because they want to manage like what you're eating and making sure that you don't have issues with basic processes like the Valsalva maneuver. So if you see this particular symbol up here at the top, this is a slide that you should make sure that you go back and review. So I want you to make sure that you know that as part of the respiratory epithelium, we will have a number of different cell types associated with it, including the outer mucosal layer that you see here. Embedded within the mucosal layer, we have these goblet cells, and these goblet cells, again, are ways in which mucus, immune molecules, and others get released. Under this mucosal layer that contains these goblet cells, we'll have the submucosal layer down here at the bottom. And then on the outside, we'll actually, so this is where air would be going through. And then on the outside, we will have, in different parts of the respiratory tract, we'll have rings of cartilage, which are known as hyaline cartilage. That in the next diagram. So this is a slide that I want you to make sure that you go back, spend some time and review to make sure that you know the different layers that we find within the respiratory epithelium. So this gets into the bronchi and the bronchioles. So in the bronchi, so this is the trachea, and this has those hyaline cartilage rings in sort of this pale green. And then we get into the left and the right bronchi, one to each lobe of the lung. And as we go down farther and farther, one of the things that you will start to notice is that eventually at the level of the bronchioles, we no longer have these cartilage surrounding them. So we no longer have the structural support that we did up here at the level of the trachea, nor at the different bronchi, the left and right bronchi. And at the level of the bronchioles, we only have the normal lining that we would have, sort of this type of lining. And in addition to that, as we get down to the levels of the bronchioles, we'll also see that they actually have this ring here, which is in the bronchioles, the smooth muscle. So this smooth muscle is capable of contracting just like it would anywhere else. And the muscle contracting actually is the cause of asthma. When you can't breathe, it's because the bronchioles in your lungs have all constricted. And when they constrict, it's much, much harder to force air down into your lungs at the level of the alveoli than it is when they're wide open. Of the smooth muscle in the bronchioles cause things like asthma and breathing difficulties overall. Any questions on this before we go on, or what you need to make sure that you know? Okay, so another slide for you to make sure that you spend some time on. I'm not a big fan of numbers, so if you're wondering, should I know like the number of orders of branching? Not really. Just for your own information, there are 23 orders of branching. It splits off 23 times until it gets all the way down to the alveoli. But when we get into the conducting zone, there are a couple of features I want you to make sure that you go back and review. So the trachea splits off into the right and the left bronchi. These are not identical, so they're slightly different. So the right bronchus is actually wider and shorter and more vertical than the left bronchus is. Okay, so it's actually different structurally. Each of the bronchi then goes into the lobar bronchi. There are 3 on the right side, 2 on the left. And again, this actually helps you pathologically to identify the right and left lungs, because the right bronchus is different than the left bronchus, and then the bronchi, they split off into different numbers whether you're on the right side or on the left side. So again, make sure that you spend some time looking at the different bronchi that help you to identify if you're in the right lung versus the left lung, and make sure that you understand the overall topology of having smooth muscle, no cartilage by the time you get into the bronchioles, which are further down in this conducting zone. From the bronchi to the bronchioles, lots of structural changes. I've already mentioned the most important ones. The cartilage rings eventually disappear completely. Cartilage is completely gone by the time you reach the smallest of these, the bronchioles. The epithelial cells also tend to change from being pseudostratified, columnar, to being more flattened or cuboidal. The goblet cells that we saw in the mucosal layer are almost gone and become very sparse by the time you reach the bronchioles, and the relative amount of smooth muscle increases. So again, all things that I mentioned to you before, just in sort of this point form type of form, for you to go back and to review. So what this means is that this is the region that is most vulnerable. So if you have asthmatic attacks, it's the bronchi, the bronchioles, sorry, that are the ones that are most susceptible to any kind of blockage or constriction. Okay, any questions before I go on? Yeah. Yeah.
For slide 41, do we just have to know like the relationship between like asthma and skin muscles? That's all I think.
Yeah, I think so. I think it's just to get the same point across that's on slide 43, which is the amount of smooth muscle increases in the bronchioles. Those are the ones that constrict. That's what's responsible for asthma. Any other questions or things I can clarify for you now? Okay. So ultimately, what we're trying to do is every time you breathe in, it goes down your trachea, it goes into your bronchi. Those are kept open all the time by the hyaline cartilage rings. They prevent them from collapsing. Eventually, they work their way down into these little bronchioles. We see less and less cartilage associated with them, almost all smooth muscle. But eventually, if we're really lucky, we get them into these little sacs, which are known as alveoli. And these alveoli are the ones that actually exchange gases, the air that we breathe that contains oxygen and allows us to get rid of CO2 that's been built up within the body. And we have about 300 million of these alveoli that comprise most of the lung volume and is the major site for gas exchange within the lung. How are they designed to be able to do this? And this is actually the more important slide. Like, there really isn't a lot of information that I want you to take from this slide. This is the more important one that follows from this. So this is the site of gas exchange. And you can see that we have a rich blood supply in terms of capillaries, and these are all capillaries, all endothelial cells, very, very small, 1 cell thick capillaries that we just saw in the previous section. And when we're talking about the alveoli, the alveoli are the site of gas exchange. The walls of the alveoli that you see here, almost like a honeycomb shape that you have here on the left, are only about half a micron thick. And they are very, very thin and allow the air to pass through into the blood incredibly quickly. The alveoli and the capillaries, to make this process even faster, have their walls fused together. So there's very little distance separating the capillaries, the blood, and the air that's coming in. All of this is fused together, including their basement membranes. And again, the alveolar walls consist of just 1 layer of squamous epithelial cells, and these are type 1 cells. Do you remember what the walls of the capillaries were formed of? What type of cells? So those are endothelial cells, and these are epithelial squamous cells. Okay, so squamous epithelium. Any questions about what you need to know before we go on? And this structure is a classic structure in terms of showing like how the body is adapted to allow really quick exchange. There's like no distance. On one side, you have air, and on the other side, through a very, very small wall between them, then the wall consisting of the alveolar membrane, the endothelial membrane on the capillary, that distance is one of the smallest that we have. So the exchange from the gases in the alveoli and the gases that exist within the blood that's flowing by is really, really small. And it's really, really quick as a result of this really thin barrier. So these alveoli and actually makes sense with the next slide. And I will tell you ahead of time that there is a multiple choice test question related to the structure of the alveoli and the different cell types that you find in the alveoli. And I think as physiology students, it's important that you know some of these major features. This is already told for you in the previous slide that this is the squamous epithelial cells. These are the ones that make up the walls. They're the most common type in the alveoli, that little cluster of grape-like gas exchange sacs. Scattered throughout the alveoli, we have these type 2 cuboidal cells, which secrete surfactant as well as antimicrobial proteins. You guys know what surfactant is? Have you heard of that in any of your other classes? Sorry? Do they coat surfaces? It's a good guess, but it's a good guess. Any other guesses? Surfactant. Are any of you interested in pediatrics or in neonatal delivery or anything along those lines? So what the big issue is, and this actually was in the news not very long ago, was a young lady who was shot, and they had to, unfortunately, try to deliver her baby through C-section. The baby was about 22 weeks of age, so not quite fully developed. And one of the biggest issues for young infants or developing infants is that their lungs aren't developed, and it's really, really hard for them. Why they die. It's not because of issues with their heart. It's not because they can't introduce intravenously like fluids and other things into them, but they can't breathe, and they have to be assisted in breathing. They lack surfactant. In order for your lungs to inflate and stay inflated, it takes a tremendous amount of effort for you to be able to inflate your lungs. And the surfactant that is secreted by these alveoli don't exist until the last trimester of development. And as a result, premature babies have a really hard time breathing because they don't have enough surfactant. And the surfactant helps you breathe all the time. It keeps these alveoli nice and open. It reduces surface tension between the water molecules on the inside of the particular structure of the alveoli. So the alveoli themselves are surrounded by these fine elastic fibers. They also contain open pores. They allow for air to flow from one alveolus to another that's adjacent beside it. That helps to pressurize the lung completely, so it's all one pressure. It's not a high zone of pressure in one alveolus and a low amount in another. And the last thing I'm going to ask you to make sure that you know is that alveoli also contain macrophages. And these are resident macrophages that exist in your lungs to help keep these alveolar surfaces sterile. So whenever you breathe in, so if you are breathing in normally, you are breathing in bacteria along with it. But most of the time, it actually doesn't affect you. So the bacteria that you breathe in will be digested by macrophages. In addition, antimicrobial proteins will help to fight off any viruses, bacteria that you happen to breathe in as well. So this is the same just shown to you in a different way. And what I'm going to try to point out are the different cell types that are available for you. The vast majority of the cells, so here's one alveolus here, here's another alveolus here, here's another down here, and here's another, and you can see that they're all interconnected. We see tiny little pores that connect one alveolus to another. Most of these consist of this type 1 squamous epithelial cell that you see here in this sort of pale color. It looks like a soccer ball, in other words. And once in a while, you'll see these pale blue cells that represent the type 2 secreting, surfactant secreting cells. I think actually it might be in green here. yes, it's in green. The type 2 surfactant secreting cells that you have interspersed between them, and then you have these macrophages. Your lungs have to be kept sterile, in other words. Okay, and those again are the major features that I wanted you to make sure that you know. Here is the oxygen, carbon dioxide on one side. Here's the blood flowing by on the other, a very, very thin membrane separating the two, and this is the alveolus. Any questions before we go on? Anything that you want me to go back and re-explain, or you're not sure what, if you need to know something, now's the time to ask. Okay, so in terms of the lung anatomy, again, this goes back to that previous diagram. Left lung, right lung are not equal. They're very different. The bronchi are different. The number of lobules are different. And this is, again, recapitulated here. The left lung generally tends to be smaller. There are two different lobes separated by an oblique fissure running across it. The right lung has three lobes and has two different fissures, one oblique and then one horizontal. So there is a difference between right and left lung. Number of lobules is different. Number of fissures is different. The right and left bronchus are also different. All the anatomy and the number of segments, etc. It's really the bullet points that I'm more concerned about. Okay, so you can pathologically, like on a dissection, on a vivisection of a cadaver, actually identify differences between right and left lungs. Okay, so you don't have to know this particular diagram or this particular slide. It's just again to give you some idea about the blood supply to the lungs. Themselves are tissue. They also require blood supply separate to them. So yes, you have blood flowing through them to get oxygenated, to get rid of CO2, but you also have to have blood supply to keep the lungs themselves alive. And this pulmonary circulation that supplies the lungs is actually really different. So it's a low pressure system, but also high volume system, because the pulmonary arteries are very different than they are in the systemic circulation. They're different than what you would find in terms of the rest of the body. We won't go into all these details now. And one major difference being venous blood is being delivered by these pulmonary arteries, and the pulmonary veins actually can carry oxygenated blood back to the heart. So that again makes it very different than it is elsewhere in the body. And again, if you want to go through this later on, if you're really interested, you can come and chat with me. I'll be happy to go through that in more detail. Okay, so what we're going to do now is talk about how you actually breathe. So we looked at the overall structure of the lungs, and we looked at things related to the microscopy of the lungs or the histology of the lungs. We looked at how the lung changes from the trachea, the bronchi, all the way down to the bronchioles, the lack of cartilage, the increase in smooth muscle, how that might be a problem for things like asthma. And now we're looking at how you actually make use of this lung. And again, of course, you're also going to look at the alveoli and make sure that you know all the details of the alveoli. So I want you to get like a really good mark on this section of the course. So we're going to look at the overall relationship of the lungs and some of the physics behind that. In your programs here, you guys all take physics, right? Really good at physics. So we're going to go through some physics. Yes. Physics is good. So just some basic physics to talk about. So right now, when you're sitting out here, you're sitting at sea level. So we're not at an elevated, we're not at elevation, we're not above sea level, nor are we below sea level. We're right at sea level. At sea level, anywhere in the world, we call this 1 atmosphere of pressure. And 1 atmosphere of pressure is equal to 760 millimeters of mercury. Okay, and again, it's something that hopefully you've learned in your physics classes, right? Common knowledge. Yes, no, maybe, not common knowledge? Okay. So I'm going to keep it really, really simple. So when we are breathing in, we actually have to change, we can't change the atmospheric pressure out here, right? Unless you have divine powers and you can change the atmospheric pressure at will outside of you, you can't do that. Air into and out of your lungs, you need to be able to create a pressure gradient. You can't change the pressure out here, but guess what? You can change the pressure inside your lungs. And you have a number of different musculatures that actually allow this to occur. So if there's 760 millimeters of mercury out here, and let's say just for example, you have 2 millimeters of mercury inside your lungs, which way is the air going to move? So it's going to move in, right? So there's higher pressure out here, lower pressure in here, there's a gradient, air will move down that gradient. So it's the same idea. So can you change the size of your lungs? And by changing the size of your lungs, can you change the pressure in your lungs to allow the inflow and the outflow of air? You can't do a single thing about 760 millimeters of mercury out here, but you can change the size of your lungs and the pressure inside of your lungs to change the pressure gradient. And that's the general principle that we're going to look at. A lot of you look confused. Confused? Kind of? Hopefully not too confused. So I'm going to go through and take a look at some specific elements. Keep this all in mind. We're looking at pressure changes relative to 1 atmosphere or 760 millimeters of mercury. So we can change the intrapulmonary pressure. This is the pressure in the alveoli. And again, we won't worry about all the details, but this is one of the pressures that we can actually measure and that we can take a look at. That's inside your lungs. And there is an intrapleural pressure, and again, you don't have to worry about the details here, which is the pressure in the pleural cavity, and this always is a negative pressure. And again, don't worry about all these details, they'll become more clear in the next few slides. So this is a slide that I want you to be aware of. So I told you earlier, we need to be able to change the shape of our lungs to change the pressure. In fact, your lungs normally, by themselves, have this innate capacity to want to change their shape and their size. So this gets into this intrapleural pressure. That negative intrapleural pressure is due to 2 different things. You have 2 forces that actually want your lungs to collapse. You've heard of like having someone's lung collapse, right? So someone's been running along, they get jabbed in the chest, or they get a really hard blow to the chest. If you ever watch like like really physical contact sports, like you get shot in the chest, that's enough to cause your lung to collapse. And one of the things that happens is, why does that occur? It's because the lungs themselves have elasticity. Remember those fine elastic fibers that are on the alveoli, outside of the alveoli? Their natural tendency is actually to collapse. They want to get as small as they possibly can inside your thoracic chest. You also have surface tension. There is fluid on that. So if you've ever seen surface tension on fluid, that actually makes fluid kind of ball up. If you ever see like a spill, that doesn't vaporize and go all over the place. It tends to stay together in globs, right? So if you spill something on the table, you'll see globs of fluid. It's not just one whole sheet. They want to collapse in on themselves and create this alveolar surface tension, or surface tension of fluids. So these are causing the lungs to collapse. Your lungs are wet. It wants to collapse. Your lungs are also elastic. It wants to collapse. There's also an outward force. The outward force is, if you were to actually puncture lungs, your natural tendency is for your chest wall to go out. And so you have this force that makes your chest wall, the rib cage inside your body, actually wants to like jump out. It's like, really happy to see you. I love you. Here we go, right? And it's this elasticity of the chest wall that actually is coming out. And then the lungs that are inside the chest wall want to go in. And so if you combine the 2 and they're stuck together, you have one thing wanting to go out, and the lungs that are inside and stuck to that chest wall, they want to collapse in. So we have these 2 different opposing forces, and I want you to make sure that you are aware of these. In fact, one of the things that happens is that we do get pathologies where we have a lung collapse. This can happen if you get a plugged bronchiole. You could have a wound where air is introduced into the pleural cavity. This is known as pneumothorax. Again, you actually have to take the air out of the lung. So normally there is no gap in air. Your lungs can't attach, and as a result, until you remove this pneumothorax actually is very, very painful, and you feel like you're drowning. You can't breathe, even though you're trying your best to be able to do so. So this could be because you get hit. It could be because you get stabbed in the chest, but it can also occur spontaneously. In a very small number of cases, you could be sitting here right now, and then suddenly you might have pneumothorax. You might have a condition where your lung starts to collapse inward. You could also have what is known as a pleural effusion. If you get fluid in this space, any kind of fluid in this space, that can also, through pleural effusion, cause your lung to move away from the thoracic wall as well. So again, we need to be aware of these different situations. So what we're going to be looking at is your lungs have these two different things, one to collapse in, the other one to expand out. Can we make use of that to change the volume of our lungs, and by changing the volume of our lungs, change the pressure in our lungs, and by changing the pressure, create a gradient compared to the outside world around us that's always constant, always staying at about the same level. And this gets into inspiration as well as expiration. And these are mechanical processes that go on where we are changing the lung volume, making use of the fact that our chest wants to expand out and our lungs want to collapse back in. There's elasticity built into it. And the very simple premise is that if you can change the volume of the thoracic cavity, your rib cage, and the lungs that are attached to your rib cage, if you change the volume, you will change the pressure. Simple physics, right? Okay. I know you guys secretly love physics. I know you go home late at night. This physics, I can't get enough of it. Let's try to do another integral and another differential equation. So these volume changes equal these pressure changes. And these pressure changes that result also cause gas to flow inward until there's no gradient across them. Hopefully that idea or this principle is a really easy one for you to follow along. And this relates back to, again, really simple chemistry slash physics slash whatever you want to call it. And this is in this idea of pulmonary ventilation, the physical movement of air into and out of the lungs depends on a couple of different laws of physics related to Boyle's law that we're going to talk about, related to Laplace's law of compliance, as well as the resistance to airflow. Now, you don't need to memorize all of these, nor do I expect that you will be able to tell me Laplace's law in terms of a formula, but I do want you to know, for example, the next slide that is related to Boyle's law, which for our discussion here today is the most important one. So this slide here, with where it says that it's really important, it's really the next slide that's related to Boyle's law that is the most important one for you to make sure that you know. Boyle's law is really simple. You guys have all learned this law before. So P1 V1 equals P2 V2. Remember that law? P1 V1, P2 V2. Another way to say this is that the pressure is inversely proportional to the volume. Okay? So let's take a look at Boyle's law related to ventilation. This is your normal size lung, okay? So if this is the normal size lung and the number of molecules that are inside it, the number of little dots that you see here that are causing the pressure inside this chamber because they bounce around, and as they bounce around, they create pressure every time they hit the wall. You can see that in this size container, this one molecule will cause this much pressure associated with it. It hits the sides of the walls like 4 times, okay? So it hits it once here, then it hits it here, here, and then it hits it here before it begins this process again. Now let's say we suddenly have increased the size of the container. We have not changed the number of molecules. It's identical. It's the same number of molecules as we have up there, but let's say let's increase the size of the box that this gas is found in. So this molecule that was bouncing around and creating pressure on the sides, do we still have the same amount of pressure? I've increased the size of the container, increased the volume. What has happened to the pressure being exerted by this one molecule? It has decreased. It's inversely proportional. So pressure is inversely proportional to the volume. You increase the volume, you decrease the pressure. That's what that inverse relationship means. So if we do the exact opposite, now let's say I make the container smaller. I keep the same number of molecules in there, but now the size of the container itself is smaller. You can see that one molecule now will bounce around more, exerting more pressure. So the smaller the size of the container, the smaller the volume is, the greater the pressure is. Now imagine that they're your lungs. So I expand the size of my lungs, which is what you do when you breathe in. So inspiration is have you increased or decreased the pressure? I've decreased the pressure in my lung, allowing air to flow in. This is making use of Boyle's law. When you are breathing out, your lungs are getting smaller again. The volume of your lungs is getting smaller. The pressure inside your lungs is increasing. Volume decreases means pressure increases inside your lungs. You breathe air out because the pressure here is now higher than the pressure out here, 760. Make sense or no? Kind of? So again, make sure that you understand Boyle's law and that they're inversely related to this. So a couple of other things, and this is the same diagram that's on the same slide after this one, which is if you look at the mechanics of breathing, when you are looking at inspiration, when you are breathing in and you are changing the size of your lungs, increasing the size of your lungs, increasing the volume means you are decreasing the pressure by Boyle's law, right? You've increased the size of your chest wall, increased the size of your lungs that are attached to it. Of doing this is actually an active process. This means that in order for your lungs to expand, the inspiratory muscles have to be able to contract to cause thoracic volume increases. The lungs which are attached to the thoracic rib cage are stretched, and the intrapulmonary volume overall increases. So the volume increasing means that the pressure decreases, and this intrapulmonary pressure drops, and because the pressure is dropping, air can now flow in. Is that okay for everyone? So these are the main points. They're summarized again for you in this slide here. So this is sort of the inspiratory muscles that are involved. So in inspiration, it is an active process which requires active muscle contraction. So your diaphragm actually descends, rib cage rises because these muscles, the external intercostal muscles, are contracting, and when they contract, the diaphragm descends, rib cage rises, increases the size of this thoracic cavity. The lungs which are attached are stretched, the intrapulmonary volume increases. By Boyle's law, this means that the intrapulmonary pressure inside your lungs must drop. The pressure out here at atmosphere is higher, and therefore it flows into your lungs as a result. Any questions about what you need to know on this slide? So would you be able to tell me like the major things that happen during inspiration? Which muscles are contracting? What is happening to the intrapulmonary pressure? What's happening to the volume inside the thoracic rib cage? No, maybe? I can guarantee you'll get questions related to this. Related to this is also the mechanics of breathing when you are breathing out, the exhalation, the expiration. This is very, very different though. So whereas breathing in, inspiration is an active process, quiet expiration. So what you're doing now is called quiet expiration. The expiration is. Okay, active exhalation. When would you do that? Exercising. So as you're sitting here right now, you are quietly inspiring and quietly expiring air in and out. This is not an active process. All you are doing is the inspiratory muscles are relaxing. They're no longer contracting. The thoracic cavity volume decreases because the lungs are elastic. Remember I told you their natural tendency is to get as small as possible. The thoracic volume decreases, elastic lungs recoil, the volume decreases. By Boyle's law, this means that the pressure inside your lungs must be increasing, and therefore air flows out of your lungs down the the pressure gradient until there is no more pressure gradient left. Forced expiration, though, is a very active process. I do want you to make sure that you know that it is an active process where it's forced or active expiration. In order for you to be breathing out more effectively, you need to actually use the abdominal muscles and then the other rib cage muscles, not the external, but the internal intercostal muscles, so that when you're breathing out, you actually collapse your rib cage in more by contracting the internal intercostal muscles. And again, this is all summarized for you in the next diagram, which shows you the same thing. Everything is just basically relaxing. It's going back to its normal state. Pressure is rising, and then in active expiration, again, we have other muscles that are being involved. Internal intercostals are contracting, as well as having abdominal muscles coming into play as well. Any questions on this before we go on? Okay, so a couple of physical factors. Again, just for your own information, we won't go into this in a lot of detail. Your muscles actually consume a lot of energy. It may seem like you're not really, but part of the reason why premature infants actually die, part is they can't do efficient gas exchange, but the other part is it takes a lot of effort, a lot of muscle activity to actually breathe in and breathe out. It may not seem like a lot for you right now, but for older individuals, it really is. And for young premature infants, it really is as well. And this energy has to overcome airway resistance, alveolar surface tension, and that involves that surfactant, more alveolar surface tension, and then the lung compliance or elasticity. Again, this is just for your own information. You don't have to memorize all of this. The resistance is usually pretty insignificant because overall it's like breathing through like your hands. There's like, the airways are so large that there's almost no resistance. They're very large diameters in the first part, and by the time that you get to smaller and smaller branches, because you have so many of them, it's like blowing through a whole bunch of straws of the same size. So eventually, the resistance disappears by the time you get to the terminal bronchioles where gas exchange occurs. This, however, I would like you to make sure that you know. So where does airway resistance increase? So airway resistance can increase if you have obstruction of the bronchioles, so things like asthma. This will prevent life-sustaining ventilation. It can occur during asthmatic attacks if you're stopping ventilation. And one of the things that epinephrine does, if those of you who are on the little puffers, the things that epinephrine does and analogs of epinephrine is to dilate these bronchioles and cause the smooth muscles to relax, and overall will reduce that airway resistance. So make sure that you're aware of things on this slide related to airway resistance at the level of the bronchioles, where you have those smooth muscles contracting and causing a cessation of ventilation. The surface tension, and this is another factor that you have to, your muscles have to overcome in order for you to be able to breathe. And the surface tension is really related to the fact that your lungs are wet, and because they're wet, there is tension between the water molecules to cause them to collapse down. So if you blow up a balloon, what's the first thing that you do? Stretch it. That's right. So you kind of loosen it up and you stretch it. And surfactant is kind of acting, being able to stretch out the balloon. It's really hard to blow up until you've kind of stretched it out. Surfactant breaks apart the stiffness of the water molecules. And surfactant is a detergent-like molecule, a lipid protein complex. It's produced by those type 2 alveolar cells that we looked at earlier, and it is something that reduces the tension of alveolar fluid. And again, it's that insufficient quantity in premature infants that cause them to have all those problems breathing. And until they produce enough of their own. You actually have to supply artificial surfactant to them in the hopes that it will help them to be able to breathe. And so this gets back into the importance of understanding the physiology. So I'm not going to ask you all of this about lung compliance. This is the third factor: airway resistance, surface tension, and then lung compliance. You have to overcome all of these whenever you're breathing in. And I'm going to tell you right off the top that the only thing I want you to really take away from this is that different types of damage, like fibrosis, reduced production of surfactant, or decreased flexibility of the rib cage will all reduce lung compliance. And this is something that becomes a problem for individuals when they need to be able to breathe effectively. So you might have fibrosis because you had an infection of your lungs at some point in time. Hopefully no one has reduced production of surfactant. Normally you wouldn't. If you're a heavy smoker, you will have reduced production of surfactant. Decreased flexibility of the thoracic cage. What do you think causes that? So you might have had a lot of trauma to your chest. That's one possibility. What's the other thing? What happens to all of us? Yeah, aging. So as you get older, you have decreased flexibility of the thoracic cage. Your lung compliance normally, as you get older, drops as well. So again, make sure that you're aware of this slide related to lung compliance. What factors are associated with decreased lung compliance? Fibrosis, reduced production of surfactant, decreased flexibility of the thoracic cage, all related to decreased lung compliance. I'm going to go through this one slide really quickly to give you an idea of what's happening here, and then I'll take you through the spirometry traces, and then we'll take a break. One of the things that we need to know, and you can actually get tested for at your family doctor, is a pulmonary function test or a lung performance test. In the old days, you used to be hooked up to what's known as this spirometer. So there's this big bell jar that goes up and down as you breathe into it. So you're connected to this, goes up and down. It moves a pen, and this pen will measure out the overall volumes of the amount of air that you're breathing in and out. And this is what is known as spirometry. It tells you a lot about the different lung volumes, and this is what a spirometry trace would look like. And there are things on the spirometry trace that, as physiologists, I need you to be able to know. Okay, so if you're ever looking at an elite athlete and you're looking to see what their lung performance is like, and you see them kind of hooked up to a machine, their nose is plugged, and they're busy cycling and they're collecting carbon dioxide, they're also doing this spirometry trace as well. So what happens is you are asked to breathe in normally and then breathe out normally. So this is in and then out. Okay, so you've breathed in, you've taken you've inspired, then you've expired air, and then you're asked to take in the deepest breath that you possibly can, and then it goes all the way up like this. And then you are asked, as quickly as you can, breathe it all out as quickly as you can, and this is what happens. And then you're asked to go back to breathing normally. So breathe in normally, breathe in as fast as you can, breathe out as fast as you can, and then go back to normal breathing. This normal breathing pattern is known as the tidal volume. For most individuals, it's about 500 milliliters of oxygen that you breathe in and out all the time. You're sitting here right now, your tidal volume is about half a liter of oxygen. Every time you breathe in, half a liter. Every time you breathe out, about half a liter. Depending on whether or not you are an elite athlete or not an elite athlete, the overall size here will be quite different. But that's not the most important thing. This measure where you're doing the deepest breath in that you possibly can and the most forceful expiration that you possibly can, where you're using all your muscles. Breathe in as hard as you can, breathe out as hard as you can. You're using active inspiration, active expiration. This is known as the vital capacity. Again, the volume not being so important. It's not a standard volume. If you're an elite athlete, if you're older, if you have lung disease, these will all be different. But I want you to know what the vital capacity actually represents. I'm going to tell you again straight off the top that on your, you will actually have a diagram related to being able to tell me about the different parts of this trace. So again, I like being really upfront. So you need to make sure that you're clear on this. The other thing that you're going to see here is this volume here. For most individuals, it's about 6 liters. Your lungs, in other words, can hold about 6 liters of oxygen or air or gas. And this total lung capacity, is it something that you can measure on a spirometer? Yes. So you can measure tidal volume, can you, on a spirometer? Yes, I've just shown you that you can measure tidal volume on a spirometer. Can you measure vital capacity on a spirometer? I can. So this thing in black says that I can measure this on a spirometer. Can I measure total lung capacity on a spirometer? I can't measure it directly. It's something that we have to be able to infer. I can measure anything in black that you see here is something that you can measure on the spirometer. It leaves a trace on that rotating drum. It leaves a trace electronically if you're going into a modern GP's office. But I can't actually measure this total lung capacity. And part of this is that your lungs can never actually really be empty of air. Like, it would be disastrous. Like, if you were able to squeeze out every last molecule of air that's in your lungs, you wouldn't actually be able to inflate your lungs again after. Because remember, it has this capacity to shrink. There's surface tension in it, and it's like having a balloon that was full of water and you've just collapsed it down. It's going to be really hard to blow it back up. And we need to keep some air in there so that the next breath is easy to inflate. So we can't actually measure total lung capacity. And this is because this volume that we have here, this residual volume down here in green, something that we can't measure, actually keeps our airway open. So can we measure total lung capacity directly? Not directly. Can we measure residual volume directly? And not directly, but we can make some inferences about it. Question.
So the wave there is the tidal volume.
The wave this here? Yeah. So this is in, out, normal breathing. That's tidal volume. Other questions? Yes. Yep.
Maintaining their breath and going really deep into the water as far as they can go. How do they practice, improving their total lung capacity?
They don't. So if you're a deep, if you're going diving deep, and there's a difference between holding your breath and diving deep, as deep as you can go into the water. So there's a couple of things that they do. So one thing is they load up on oxygen as much as possible, and we'll talk about why they do that later. The other thing that they do is they will not have a full stomach. So the emptier your stomach is, the bigger your lungs are. That affects like the capacity of your lungs. They normally do this in as cold weather as possible, and as you know, like the farther you go down, the colder it gets. It's actually better for their breathing and their lung capacities. But again, overall, it's not really to do with increasing your lung capacity. You can't. Like you can train for getting better oxygen loading and offloading, but you can't really change the size of your lungs. Any other questions before we take a break? I think now is actually a pretty good time to take a break, and then we'll sort of wrap things up for this evening.
My coach once told me that you shouldn't be eating an hour before. You have good training session and an hour after. And is that why? Because your lung capacity is higher and you're able to do more vigorous exercise, or just like a follow up? Okay. Okay. Like tennis, for example. Like I couldn't, she wouldn't let me eat an hour before.
Yeah, I think there's this idea that change in what you go to adjust the system. The activation of the nerves.
Uh-huh.
Adjust the blood flow.
Yeah. But I know even like a couple of times where I eat, like I would feel nauseous and I would feel like food would be coming. Yeah. Okay. All right. Well,
I just have a question. Do you teach any other course except is there a way you can come back again and no? So what other courses do you teach? So would you teach immunology as well?
We do that in second year.
Which campus are you?
St. George.
Bro, I'm trying.
Okay. I guess before we officially get going, how do you want me to help you prepare for your upcoming term test? What should I do? Okay, sure. So just some review jot notes. Okay, done. I will post that for you over the weekend. So I'll also have a couple of advanced questions for you, so that it'll look pretty much like the kind that you're going to get on the upcoming term test in advance of the term test. Okay, so I think we have a strategy for how to get you ready for the upcoming term test, and we'll be posting the sort of outline jot notes that we did last time, and a couple of questions in advance of the actual term tests for you to practice, discuss, etc., to make sure that you're ready. I'm hoping with just having 2 lectures, having some guidelines for what you should be knowing from each of the slides will help you study more effectively this time. I'm hoping that your class as a whole will be able to get sort of a 75% average, to be quite honest. If you don't reach it, I'm hoping that based on your last sort of term test that you'll be able to get sort of around a 75. These spirometers are not only useful for the physiological measurement of different lung volumes. And by the way, if you're wondering, why are some of these things called volumes, like inspiratory reserve volume, tidal volume, expiratory reserve volume, residual volume, versus other things that are capacities? If you are using a spirometer, a capacity is actually the combination of more than one lung volume. So if I add together the inspiratory volume, the tidal volume, expiratory, residual, then I get a capacity, like the total lung capacity. And that's the difference in why we name some things volumes. It's just like one measurement, and the capacity means that multiple volumes are added together. So anytime you see a functional residual capacity, it's the expiratory reserve volume plus residual. And again, you don't have to worry about those details. It's just for your own information in case you were wondering. Spirometers, though, are not just there to measure these volumes and capacities. They're used clinically, and they're still used clinically today to be able to diagnose things like obstructive pulmonary disease. If you have increased airway resistance through bronchitis, for example, how quickly you are able to breathe in and out, and the forced expiration, like how quickly that occurs. You're able to tell the difference between obstruction versus restrictive disorders on a spirometer. And restrictive disorders might be because you have physically damaged your lung. So you might have fibrosis, and we've seen fibrosis before. That part of the lung that no longer is elastic, no longer can recoil, can't take part in gas exchange, means that it doesn't allow for as much air to be moved in and out. We don't see a lot of tuberculosis anymore, thankfully. It's a terrible bacterial infection in your lungs. It takes an awfully long time for you to actually clear up this kind of lung infection, and it's very, very contagious. So if you've ever wondered why really cares about cases of tuberculosis. It's really infectious. If you get tuberculosis, it's very easy to get tuberculosis. Like everyone in here is kind of coughing, right? But if you have tuberculosis and you're coughing, it can actually infect your lungs, a normal healthy person's, really, really easily. So if we're in an enclosed room like this, someone in this classroom has tuberculosis, okay, and everyone stopped coughing all of a sudden, and they were to cough, it's likely that about a third of us would actually contract tuberculosis. So it's a very, very bad disease to have. It's something that can be measured on a spirometer. It is a type of restrictive disorder, and these spirometers are useful not just for measuring normal physiological volumes and capacities, but also the differences between different types of diseases. So this gets into that dead space. There are different types of dead space. There's an anatomical dead space and an alveolar dead space. And then we also have this value here, which is this residual volume down here in green. And all of this contributes to the fact that we need to have some air left in our lungs in order to continue this process of being able to breathe in and out. But this also means that this doesn't actually take part in gas exchange. It's there and it's just moved around different parts of your respiratory tree. Again, some other pulmonary tests that can be done. There is this minute ventilation. It's not minute, it's minute ventilation. You measure the total amount of gas into and out of the respiratory tract in 1 minute. There is this forced vital capacity. It's not just take the deepest breath in that you can and then breathe out as much as you can. It's breathe in as fast as you can and then breathe out as fast as you can. That is something, and I can't remember who asked this question, that is something that you can change. That's due to changes in your muscle. You can change like how quickly you move things into and out of your lungs, but you can't change the size of your lungs, okay? So this forced vital capacity can tell you about the lung muscle function, and the same thing with the forced expiratory volume as well. And all of these reductions could be a result of fibrosis or other types of restrictive diseases like tuberculosis. And you guys really like math, physics, everything else. I'm really hitting you with all this math and physics today. So here's another thing that I'd like you to make sure that you know. Just like with the cardiac output, do you remember what the formula was for cardiac output that you need to know for Monday? So we have cardiac output equals the heart rate times the stroke volume. Normally this is about 72 beats per minute. And again, this is maybe on the order of about 150 mils or so. We can do the same thing with the respiratory minute ventilation or the respiratory minute volume. So how much air is moved into and out of your lungs is the same thing as how much blood is pushed out of your heart per minute, how much air is moved into and out of your lungs per minute. And what we can do there is calculate this. The ventilation is equal to the frequency of breathing times the tidal volume. So it looks really similar, right? The heart rate is like the frequency of breathing, the number of beats per minute, the number of breaths per minute. And usually it's on the order of about 10 to 12 breaths per minute. And you guys remember the tidal volume? What was it? Well, it's there. So it's 500 mils, and remember on this diagram, this is that tidal volume, that 500 mils that you see there. What it means is that when you're sitting there doing nothing else, you are moving 6 liters of air into and out of your lungs per minute. That's actually almost the same as your cardiac output. You are moving about 5 liters of blood out of your heart per minute, and you are moving air out at about 6 liters per minute. So they're almost equal. This is, again, going into something that's a little bit different. This gets into, that's just all the air that's being moved and pushed around your lungs. That doesn't tell you about how much air actually gets down into the alveoli. So think a little bit about this. Why do we care about the alveoli? Don't we just care about the lungs? We love our lungs, we hate our alveoli. No, we love our alveoli, we hate our lungs. No, we love both, right? We're a very loving class. But why do we care about the alveolar ventilation rate? The air that actually gets into the alveoli. Why do we care about that? Yes. That's where gas exchange occurs. We don't really care about all the air that's moving into and out of our lungs. We care about where it gets into the alveoli, because it's really the alveoli that take part in gas exchange. And so one of the things that we need to take into account is that we can look at the ventilation, the respiratory minute ventilation, but we also can look at this alveolar ventilation rate. And this alveolar ventilation rate looks almost identical. It's the frequency, breaths per minute, about 12 breaths per minute. And then we have the tidal volume, about 500 mils. But I also told you that we have this anatomical dead space, the stuff that's sitting up here, the stuff that actually doesn't take part in gas exchange, the trachea. Do we have any alveoli with the trachea? Do we have any alveoli associated with the bronchi? We do not. None of that takes part in gas exchange. So if you take all of that out and you subtract off dead space, which is normally constant, and do you guys remember what that was? Because I didn't tell you to memorize it, but I do need you to know it now. Do you remember what that was? So it was 150, that's right. So you take 500 mils of air in, but actually if you subtract out the 150 in the dead space, which is normally constant, you're really only taking in 350 milliliters that's actually used in gas exchange. Okay, so it's not as efficient as we normally would like to make it out to be. And again, don't worry about all the details down here. You can actually take a look. This is one of those really old questions that used to be asked about, is it better to take a lot of shallow breaths, or is it better to take really deep breaths, lower the frequency, take deep breaths? Because of this, it's actually, you can do the calculations, but the slow deep breathing is actually the better way, the more effective ventilation than the short shallow breaths. And again, this is for your own information if you're really curious and interested in following through. And so this gets back into the last part related to this gas exchange and this lung function. So we've looked at the mechanics. We've looked at the different parts of the structure. So we've looked at the lungs, we've looked at the alveoli, we've looked at the overall changes in the composition of the bronchioles, etc. We've looked at how we change the shape of our lungs, increasing the volume, changing the pressure as a result. We've looked at spirometry, the overall lung volumes, and now we've just talked about the alveoli and the alveolar ventilation rate. Now we have to talk about this gas exchange. This is one of the big things that your lungs are actually involved in doing. And this, again, I hate to say this, goes back into physics and chemistry. So remember Boyle's law, kind of? Pressure, volume, inversely related. Do you guys remember, like a long time ago, Dalton's law of partial pressures? So really easy. You guys all feel confident about Dalton's law? Okay, this is good. I like it. So Dalton's law of partial pressure says that the total pressure of a gas is the sum total of all the individual parts of that gas. So the 760 that we're feeling here in the air around us is actually due to all these other gases that are here in the atmosphere, nitrogen, oxygen, and carbon dioxide, that combine together. Each of the partial pressures, the amount of pressure exerted by nitrogen plus the pressure of oxygen and CO2, add up to 760. And this is what Dalton's law says. Why is that important? Because we need to be able to look at composition, which is constant, of oxygen, the partial pressure of oxygen, the amount of pressure that is exerted by oxygen. Of all the gases that we breathe in, it only makes up 21%, or about 20.9%, or 21% just to round it off. And I'd like you to make sure that you know that. So this one value here is a really important value. You should also know that, relatively speaking, and you don't have to memorize this value, is by far the most dominant gas that we breathe in. We actually don't make any use of it. You breathe in nitrogen, It makes no difference to you unless you are a deep sea diver, in which case the nitrogen that's dissolved in your bloodstream has a severe impact on you when you are coming back up from the depths. Again, CO2 is negligible, it's almost zero, and then the water vapor is almost zero, all of this adding up to 760. So the 760 that we saw before, the oxygen only has a partial pressure of 160. It makes up a much smaller amount than the total atmospheric pressure that we normally breathe in. So again, Henry's law, do you guys remember Henry's law? Okay, I don't remember Henry's law. Can you guys tell me what Henry's law represents? Why might it be useful? Yes.
It's like if you have, the more pressure you exert on a liquid, the more particles of gas are likely to dissolve into it.
Yeah, so the amount of gas in solution directly proportional to the partial pressure. So the higher the partial pressure, the more that you can get dissolved into solution. So again, it makes a lot of sense that it does that. We won't worry about all of this. The most important part that I want you to memorize, which is why you have this little diagram up here, and the only part that I actually want you to memorize, is that when you are looking at Henry's law, which says that the more partial pressure you have, the greater the amount that gets dissolved. Carbon dioxide, which we have, is it more than oxygen or less than oxygen? The total amount, the partial pressure of carbon dioxide, is it more or less? A lot less, right? So carbon dioxide, which has a very, very low partial pressure, actually it dissolves a lot more in liquid, and it's 20 times more soluble. Even though we don't have a lot of it, the parts that we do have dissolve a lot better in water than oxygen does. Okay, and our bodies are composed mainly of water. So keep that in mind. This becomes an important thing later, and this is that one point that I want you to remember. Carbon dioxide is much, much more soluble in water than oxygen is. And that's the only point that I want you to take away from that. And again, we won't go into details about the alveoli and CO2. So why are we talking about all these partial pressures? Why do we care about the partial pressure in the oxygen out here at atmospheric pressure? Why do we care about all these partial pressures at all? Why do we care about the amount of gases that can get dissolved in fluid? Part of this is because we always are talking about gradients. We move things from a high to a low gradient, whether we're talking about ions, whether we're talking about pressures, whatever we're looking at, it always goes from high to low, where we have high concentrations to low concentrations, high pressure to low pressure. And so one of the things that we need to understand is that when we are talking about the movement of different gases at the lungs, the only reason that oxygen diffuses into the blood from the lungs is because there is a pressure gradient. So we breathe in oxygen at a partial pressure of about 159. By the time it gets down to the lungs, it's about a partial pressure of 100. In your bloodstream, the partial pressure of oxygen is much, much lower. It's only, by the time it gets back, it's only at about a partial pressure of 40 before it engages in gas exchange. So there's this huge gradient from 100, as it's coming back, it's deoxygenated at 100. And it's the same thing the other way. The partial pressure of carbon dioxide in your blood coming back to your lungs is much higher than it is inside your lungs when you breathe that carbon dioxide in, and the partial pressure there is only 40. So there is a gradient for it to leave. It's the same thing at the level of the tissues. The number's not important. It's just to establish that we need these differences in partial pressure to allow these types of changes to occur. You don't have to worry about the numbers that are associated with this right now. You don't need to worry about the individual values at the lungs, in the bloodstream, at the lungs, the different tissue levels, etc. So this again is just suggesting that there is a gradient from somewhere from about 104 millimeters of partial pressure to about 40, and this gradient allows oxygen to be moved in and out of tissues and in and out of your lungs. And that's the only thing that this is really showing. I don't need you to memorize these numbers. And you can see how fast this actually takes. This is in seconds. By the time it starts in the capillary to the time that it is at the end of the capillary, it's only 0.75 seconds. And that's how much oxygen can be loaded on to the pulmonary capillary as it's passing by lungs. It's incredibly fast, in other words. It takes less than a second for it to occur, and that's because there is a steep gradient that allows it to be loaded on. Again, we won't worry about the partial pressure gradients and solubilities of carbon dioxide. Again, it just mentions here, carbon dioxide is much more soluble in fluid, in this case it's plasma, than oxygen is. And that's the only point that I want you to take away because it becomes important in a few moments. Any questions before we go on? Okay. We won't worry about this ventilation perfusion coupling because it gets into these bronchioles and the regulation of the bronchioles related to different levels of CO2 and oxygen levels. And we'll skip by this slide. It's not a really important one in terms of the physiology, but in terms of this, we've already covered like how quickly this occurs because of the thinness, and hopefully it is a review slide for you. Nothing really to worry about here, as long as you remember what sorts of things are part of the alveoli. So that's important. To remember what cells make up the capillaries, that's important. And then to know the different types of cells within the alveoli, again, that's important. These other ones here, not so important right now. The next slide, though, is one that is important. It's one of those rare slides that I'm going to ask you to memorize numbers that are associated with it. I won't be testing you directly on the numbers, but it will help you not to panic when you see these again later. But it's to give you an overall amount or comparison. If you are looking at the transport of oxygen in the blood, and you are looking at the oxygen that is carried in the blood as a whole, only 1.5% of it is actually carried dissolved in plasma. So remember Henry's law, partial pressures, more pressure, more dissolved in fluid. Oxygen doesn't like dissolving. Little of it dissolved in your plasma. Most of the oxygen that's transported in your blood is actually bound to hemoglobin. About 98.5% of all the oxygen that you're carrying in your blood is found on your red blood cells. Okay, and these are the red blood cells. Packed within these red blood cells, we have this hemoglobin molecule with this iron moiety right in the middle. This iron moiety allows 4 molecules of oxygen to be bound onto the hemoglobin. And so this is the major way, 98.5% of oxygen is bound to this hemoglobin molecule. And this gets into some rather interesting concepts about oxygen, the binding to hemoglobin, and how things work. So the loading of oxygen, you've just breathed in, right? And remember, you breathing in is a change in lung volumes, change in pressures. It gets down into your alveoli really quickly. You can calculate the alveolar ventilation through that alveolar ventilation rate formula, which again, you should make sure that you know. It's down there in your alveoli. The walls between your alveoli and your capillaries are really small. It wants to get into your bloodstream as quickly as it possibly can. It takes less than a second for it to do so. There's a steep gradient, and now it's hitting the blood. And now what's happening is, as oxygen starts binding to the hemoglobin inside your red blood cells, as it's run by the lungs, is starting to happen. So as oxygen starts to bind, the first molecule of oxygen it starts to bind, the affinity for hemoglobin actually starts to increase for you. Cooperativity, sure. So that means that the first molecule is really hard to get on, but the second and third and fourth molecules are actually really easy to get on. So as soon as the first molecule binds, hemoglobin wants to bind even more of it until it gets fully saturated. And again, it's the exact opposite. As soon as oxygen is getting released at the tissue, the affinity for oxygen on hemoglobin actually decreases too. So once you've unloaded one oxygen, it's hard to get rid of that first oxygen, but as soon as you've released that first oxygen from hemoglobin, the second, third, and fourth molecules are relatively easy. And we talk about fully saturation when all four heme groups are carrying oxygen. All four possible binding points for oxygen are there, versus partially saturated when we only have 1 to 3 of those heme groups being carried. If we were to plot out the oxygen hemoglobin dissociation curve, so it likes binding oxygen and it likes giving up oxygen. And one of the things that we started to do was to start mapping out the partial pressures of oxygen, and then the amount of oxygen that was being carried by the hemoglobin, because remember, that's the major way in which we carry oxygen around in our tissues. And if we were to take a look, lungs, the partial pressure of oxygen is normally at around 100. It's about 104, but we'll round it down to about 100. Do you remember that value? So it's at about 100. And if we were to take a look at the tissue level of oxygen, when it's coming back deoxygenated back to the lungs, it's down at a partial pressure at about 40. This is what the partial pressure dissociation curve looks like. And so when you are having hemoglobin and you have like 0 partial pressure of oxygen, as you start loading more and more oxygen onto these hemoglobin molecules, you'll actually see this shaped curve. This S-shaped curve, what does the S stand for? So it's sigmoidal. It shows cooperativity. It's hard to get that first molecule on, so it stays low for a little while. As soon as you start binding, it goes up really, really rapidly. And then adding that fourth and final molecule, again, just sort of saturates this off this way. And this sigmoidal shaped curve, or S-shaped curve, shows binding versus the release of oxygen related to the partial pressure. So the partial pressure of oxygen at the lungs at about 100, this is what it looks like up here, versus the partial pressure down here at 40. You can see that the overall percentage of saturation, it's not 0 at the level of the tissues, it's closer to about 75%. Okay, so your hemoglobin is not fully desaturated. It's still carrying oxygen, in other words, even as it releases oxygen at the level of the tissues. Any questions about this before we go on? I get a lot of, what the hell are you talking about looks right now? No? So we start off in the lungs. The maximal amount of oxygen that we can possibly carry is at the lungs. We breathe in as much oxygen as we can. Your blood is leaving your lungs and is rushing toward the other tissue. It's fully saturated here at 100%. And at the partial pressure, the highest we can get it in the lungs is 104. It's right there. We get full saturation at 100. It's now going all the way to the tissues, which are down here at 40. And how much oxygen have we unloaded? We've only unloaded about 25%. Your oxygen, your hemoglobin still, as it's working its way back to the lungs, as it's left oxygen at the level of the tissues, we've unloaded only about 25%. It's only down to this level. Okay, just keep that in mind for 1 second. If you are exercising and you need much more oxygen, and we'll see how this all occurs, this will go all the way down here. Your exercising tissues in your muscles, as blood is flowing by it, will have used up much more oxygen, and you can see the level of unloading. You are unloading oxygen almost at 80%. You've lost 80% of the oxygen that you carried during exercise. So this tells you a little bit about this oxygen hemoglobin saturation. So again, in arterial blood, it's at about 100 millimeters of mercury. I don't need you to memorize this value, about 20 mils of oxygen per 100 mils of blood, 20% volume. The saturation we've already talked about, and again, we won't worry about this venous blood volume. So I'm going to talk to you about this oxygen hemoglobin saturation. There are some factors that actually increase the capacity for your hemoglobin to unload oxygen and to keep oxygen on. So there are factors that will modify the structure of hemoglobin. So temperature changes will modify the oxygen hemoglobin saturation curve. Acidity in your tissue will affect this. The CO2 levels will also affect this. And a metabolite known as BPG or 2-DPG, and I'll show you this in a moment, as you exercise, you produce this molecule, will also affect the oxygen hemoglobin dissociation curve. All of these things work to modify the structure of hemoglobin. It means that it doesn't bind oxygen quite as well as it normally does. These types of mechanisms occur in those systemic capillaries at the level of the tissue. And what they're really designed to do, increases in temperature, hydrogen, CO2 levels increasing at the tissue, and the production of metabolites like BPG, all of this is working to enhance oxygen unloading. And what it does is that as blood is flowing by, and as the hemoglobin is now encountering all of these changes at the tissue, like temperature, acidity, CO2 levels, metabolite levels, all of this is causing the hemoglobin um, binding curve to shift to the right. And again, it's really important that you remember this. If you are moving it the other direction, you are decreasing temperature, making it colder, you are decreasing hydrogen, you are lowering CO2, you are decrease BPG, actually shifts it the other way to the left. So let's take a look at this. This is the oxygen hemoglobin saturation curve that I showed you earlier. These are the sort of regular values where we have 100 millimeters of mercury, 100% saturation, which is shown right here at the top. At the tissues, normally we have about 40 millimeters of mercury, and the overall saturation is shown for you here. So this is 100% here, and then this shows you about 98% saturation, so it's off just a little bit. At 40, what you see is that it gets down to a level of about 75%, as I mentioned to you earlier. So during rest, this difference that you see between what you have at the lungs and what you have at the tissue is only about 22. So you subtract 98% saturation minus 75% saturation, roughly you get about 22% of the oxygen is unloaded. Now, keep in mind that these are factors that will change the release of oxygen by hemoglobin. And we won't worry about the Bohr effect, it's just a fancy name. Heat production, I've already talked about. I want to show you this on the next slide because it becomes important. These are all of the factors that I mentioned to you earlier. So I mentioned to you on that previous slide about the amount of oxygen that hemoglobin can carry. This is the normal curve shown to you in the solid line here. This is what would normally happen. And remember, at this particular level of 40, this is what we get in normal tissue. So we're not doing anything else. All we're doing is either increasing the temperature, as we showed in the previous slide. By increasing the temperature, we shift the oxygen hemoglobin dissociation curve to the right. And this is that rightward shift. You all see the rightward shift, right? It's moving to the right? Yes? Okay, so this is that rightward shift. It just means that the hemoglobin now is in a warmer environment. So your muscles are exercising. You've increased the temperature of the systemic capillaries. The hemoglobin now sees a warmer temperature around it. Now let's take a look at what the effect is of the hemoglobin. You're carrying the same amount of oxygen. You've loaded it up at the lungs. What is the effect locally of doing this? So this is sort of the normal unloading here at 40. So we would be down here at about 75%. If I increase the temperature, do I unload more oxygen? Well, if I go at P40, and one of the things that happens is now at P40, I'm all the way down here at about 65%. Have I unloaded more oxygen when there is a warmer temperature? Yes, because now at the same level, at this 40, I'm all the way down here. So I've actually shifted this downward by a lot. I'm unloading all this extra oxygen because of a shift in temperature. So during exercise, the hemoglobin unloads oxygen better. What other things cause a shift to the right? A shift to the right is caused by increased temperature, increase in this BPG or DPG, and this is the 2,3 biphosphate BPG. This is a metabolite that is produced during exercise, or a shift in hydrogen ion concentrations. All of these things will shift things to the right, making oxygen easier to unload. If I shift things to the left by increasing the pH, decreasing temperature and DPG, and I shift it to the left, it actually is harder for oxygen to unload. I don't unload as much oxygen as I normally would. So those are factors that I'd like you to make sure that you are comfortable with for the upcoming term tests. So again, the effect of temperature is very pronounced. So normally we're sort of sitting here in this purple line. You can see what happens. The percentage of oxygen saturation is going to be very different. So we use this as sort of the marker, 40. If we go all the way up here, we unload at about 75%. Now, if we shift it to the right, which is this red line, increasing temperature, 40, we've unloaded all the way down to about 50%. We've unloaded half of our oxygen. But if we make ourselves colder, and this is why, like, you hear about individuals that fall into a cold lake and they go unconscious, and you're able to revive them later, it's because their oxygen usage, their anoxia, actually isn't as pronounced. You're not using as much oxygen because you don't have to unload as much. So at 20 degrees, for example, you're still almost at 100% saturation. You lose as much oxygen, and here at 40 at 10 degrees, it's almost at 100%. So the shift in the left to right on the effect of hemoglobin saturation and unloading of oxygen is actually very pronounced. The same thing for pH, more acidity to the right. You're dropping the pH levels. When you're exercising, you build up lactic acid, you drop the pH down. It makes it easier to unload more oxygen. So instead of unloading the normal pH value at 75% that you see here, if I'm exercising and I'm making it more acidic at P40, if I go all the way up, it's closer to about 65. I've unloaded more oxygen because I need more oxygen when I've shifted the pH. And again, you can go back and compare. That's that line. If you draw a line straight across, you'll see that the levels of oxygen that get unloaded are very, very different depending on the pH. So during exercise in acidic tissues, you'd have a lot more oxygen being unloaded. You need that when you are exercising, in other words. Okay. So I won't ask you this because I think that we haven't actually gone into red blood cells a lot, so we will leave this. Your colleague actually mentioned this earlier about carbon monoxide poisoning that can actually lead to hypoxia. Again, part of that is on the next slide. This slide, though, I will ask you to make sure that you know. We talked about oxygen is a little different. I told you that it is roughly 20 times more soluble. And if you go back and do the calculations, the total amount that's dissolved in plasma in the fluid is somewhere between 7 and 10%. So it's roughly 20%, 20 times more soluble. Don't worry about that number. The vast majority of this, though, is actually carried within red blood cells, not attached to hemoglobin. So hemoglobin, there is a little bit that gets attached to it, but the vast majority of this is actually carried in your blood on your red blood cells in the form of carbonic acid, which is this reaction that you see down here. So carbon dioxide mixes with water in your red blood cells through the action of an enzyme known as carbonic anhydrase. It gets converted very quickly into this carbonic acid, H2CO3, that you see here at about 70%. However, the 70% that is converted by H2CO3 actually quickly dissociates into hydrogen as well as bicarbonate. So even though we talk about it's not an acid that's carried around in its true acidic form in the red blood cells. It's actually a bicarbonate form that is released. And again, the major thing is this carbonic acid is the major form in which we carry around carbon dioxide, very little on hemoglobin, and a little bit dissolved in plasma as well. We won't go through the Haldane effect. It's very similar to the Bohr effect. It's also related to how PCO2 values can actually influence the amount of oxygen that's being carried, but I sort of skipped through that in the first part, so we won't worry about the Haldane effect in this part. I will ask you to know this because it is important, and we will be coming back to this a little bit later on in the course. In your medulla, at the bottommost part of your brainstem, so your spinal cord ends, and then just before you get into the pons and the other parts of your brain, as part of your brainstem, at the bottom of the medulla, we have the medullary respiratory centers. So we have two different groups. One is the dorsal respiratory group, or the DRG. Don't worry about it being located near cranial nerve number 9. So it's looking for activation from peripheral stretch. But the other group, the ventral respiratory group, is the one that's really the important one. This is the group that's in your brainstem that generates the rhythm of breathing. If you were to damage this group of medullary neurons, you stop breathing. And we talked about 12 breaths per minute. This is the group of neurons that sits in your brainstem that sets what's known as eupnea. So eupnea is the normal, regular breathing rate. Did you get your tickets? Will you, if you get an autograph from Beyonce, will you bring it back to class? Okay. I'd actually really prefer if you went to the Selena Gomez concert, but that's okay. Have fun. Don't forget to study. So this eupnea of about 12 to 15 breaths per minute is the normal resting rate, and it's really important. And this is the group that I want you to know that actually controls your regular breathing cycles. Okay, and if you damage this, it's pretty much game over for you. This is where you're brain dead. If you've damaged this, there's no coming back. You're technically, clinically brain dead if you've damaged this group of neurons. And again, we won't worry about the overall muscles that it helps to control. And we won't worry too much about the chemical factors that influence breathing. We will actually come back to some of these chemoreceptors when we talk about the renal system and talk about pH and PCO2 levels. So this slide here, again, not for you. I'll talk about this really, really quickly because I think it's really important for clinical notes. You know, lung cancer right now is actually a really big deal. So it's still the leading cause of cancer deaths in North America. So you take all cancers, still lung cancer, right? Why is that a bad thing? Because 90% of all lung cancer is actually caused by smoking. Nowadays, it's become a relevant discussion again because people have started using those electronic cigarettes, the vapes or whatever they're called. Those are equally as bad. So far, the initial evidence has suggested that smoking is electronic or cigarettes is still bad for you. And the common types of lung cancer include squamous cell carcinomas in the bronchial epithelial cells, and adenocarcinoma, which is in the peripheral lung, and then small cell carcinoma. And this, again, all of them are bad, and some of them are worse than others. So small cell carcinoma is actually really bad because these lymphocyte-like cells that start off in the lungs actually spread to other parts of your body. So even if you remove part of your lungs, if you have small cell carcinoma, it's going to metastasize to other parts of your body. And adenocarcinoma is never a good thing, whether it's an adenocarcinoma in the pancreas or an adenocarcinoma in your lungs, not a good thing. Why, again, is this all bad? It's a behavior-related cancer. So you're giving yourself a cancer. You are choosing to give yourself a cancer by smoking. And again, 90%. Do you need to have that cancer? I don't think so. Asthma. So again, this asthma that we talked about before is this sort of inflammation, inflammatory pathways that precede this bronchospasm. We'll be talking about some of these pathways later on, and this airway thickening and the bronchospasms, where we will activate asthma by causing the smooth muscles to start constricting and contracting, also something that is clinically really, really relevant to us. Again, all of this is just for your own information. You don't have to worry about this. So even if you are really lucky and you have bypassed having lung cancer somehow, because again, if you smoke, the likelihood of you developing cancer goes up about, I don't know, I heard the last number was about 200 times, 200%. So even if you somehow get really lucky and you don't get lung cancer, this is what happens to your lung performance. So your overall lung performance, if you are a regular smoker and you smoke a pack of cigarettes per day, and you started at the age of 25, so some of you will be deciding whether or not you should be smoking once you get into the workplace, your lung performance is going to be steadily declining as you go along. Again, maybe not a bad thing. Maybe you're planning, I'm going to stop after 20 years of smoking. Well, if you stop after 20 years of smoking and you never smoke again for the rest of your life, your lung performance is already decreased at a level that as you continue to age, you'll never recover function. If you are at the stage where you have been smoking and you've been smoking for a number of years and it's actually caused lung function compromise. So those individuals that you see carrying around these little oxygen containers, it's too late for them. They're not going to make it much more than a couple of years. And I hate to say that, it's because once you've reached a certain level of disability, you can't recover. And compare this to someone who's never smoked. I told you earlier that your lung performance as you get older is going to be compromised. And again, it's going to reduce to about 75% by the time you're 75 years of age. But look at the overall rate of decline. You're still having a lot of lung capacity left, even at a very, very old age. So when you're talking to your parents, your friends, your colleagues who are smoking, and they're putting themselves at risk for decreasing respiratory performance at the very best, or putting themselves at risk for cancer at the very worst, tell them maybe it's time for them to stop at an early age, before it becomes too late, before things get worse in terms of cancer or everything else. Should I have a really quick question and then we'll wrap things up? People who smoke once every month. Yeah, I'm not, honestly, I don't really know. I don't know. Okay, so a couple of things before you go. So over the weekend, I will be populating your your D2L site and the Facebook page with sort of a summary jot note for you to study, like study this slide, study the particulars on this slide. Make sure that you're on time on Monday. There will be a couple of practice questions as they will appear exactly on the test for you, and I think I agreed to do that for you this time around. I am expecting you to do well. Spend your time going over these two lectures in as much detail as you can over the next few days, and let's hope the Raptors actually win tonight. Okay, I'll see you all next Monday, and there will be a group activity next Monday.